So good morning, everyone. So I'm Will. Today we're going to be going through everything to do with gene expression for your A-level biology. So we'll look at some stuff on mutations, we'll look at stem cells, we'll look at things like the polymerase chain reaction, gene therapy. So it's quite an interesting session today. So we'll try and tackle all of those topics. So today we're doing something slightly different with the way that you interact with the session. So we're using Slido today. So I'm sure that it would have been on an email or something like that. So today our Slido code is, so if you go on Slido and you type in this code, which I'm about to write on the screen, then you'll be able to do all the polls and things that I flash up. So it's number X976. So I'm just going to give you a couple of minutes just to type that into Slido. So if you type in Slido into Google, it will come up. So that's like this, Slido. And then when it asks you, on the home page, it should ask you for the code. And if you type in this code, so you'll just need to type in X976. And then when I flash up a poll or something for you to interact with during the session, then you'll be able to do that on the app. So I've got it up on my phone as well, so I'll make sure it's all working as we go through. So just a couple of minutes now, just to try and to sort that Slido code out. So if you get it up on your phone or something like that, and then you won't need to switch between the Teams slide and the Slido website. So it works a lot like Kahoot if you've done that in class already. just another minute to sort that out. So once you've joined, you should get a screen that says there are no active polls at the moment, and that's where you need to be. So the chat box on the Teams link will still be open. So if you've got any questions, ask them on there, and then we've got some people that are moderating the chat, and they'll be able to reply back and answer any of your questions. And then at breaks, so at lunch or just after lunch, and at the end of the day, I'll try and tackle a few of those questions, but I won't be flipping back and forward like normal. So we get started now. So the topics that we're covering today, so we're going to look at mutations first. Then we're going to look at the regulation of transcription and translation. So we'll look at things like methyl groups, short interfering RNA, and the different ways in which we can kind of change the way that genes are expressed in our body. Then we're going to look a bit at gene expression and then cancer. So we've got quite a bit on cancer, which should be quite interesting. And then finally, probably after lunch, we'll look at gene technology. So things like polymerase chain reaction, short variable number tandem repeats, DNA profiling, gene probes, everything like that. So I understand that quite a lot of these topics are quite hard. So when we're looking at kind of processes, I'll try and go over them a few times and make sure I go slowly so they're nice and clear for you all. So like always, we'll have past paper questions at the end of each of these sections. And then with the Slido, there'll be certain points where a slider will come up on the screen. And then if you go on your phone or your laptop or whatever you've got the Slido up on, you'll be able to answer that. And then we'll get kind of graphics of the numbers of people that have kind of answered each question. So we get started now. So our first topic today is mutations. So a few learning objectives for the mutations. So want to know the different types of mutations and how gene mutations occur. And then we'll look at how mutations can affect genes. So obviously our genes are codes of proteins. So if we have mutations in our genes, that's going to affect the codes for those proteins. So that's going to have a kind of downstream effect on the proteins that our cells produce. So like always, this presentation is quite general and should cover all bases for all of the different exam boards. So here are the AQA here are the AQA specification points for the gene section, and OCR. So we've got kind of bits on the different types and the effects that these might have. And then for the Edexcel, we've got another learning objective there. So this is relevant for all of the different exam boards. So to start off, so mutations are changes to the order of the DNA sequence. So they're random changes. They can either have no effect, so silent mutations. So you'll have looked at kind of in year 12 the fact that in our DNA we have the triplets of bases which code for one amino acid, and then we have quite a lot of overlap in the different triplets. So the fact that we can have two triplets that code for the same amino acid means that sometimes a mutation might just change from an amino acid that codes for one protein to another triplet of bases that codes for the same amino acids. So a mutation like that would have no effect. Could have a harmful effect. So in the third section of today, we'll look at cancer, and so cancers, mutations are significant in cancers, and then on rare occasions they can have a beneficial effect. So we'll look at one example of how a mutation can affect an individual developing malaria. So that's in the red blood cells. So mutations are the drivers of evolution. So random mutations, if they have a beneficial effect, then they'll be selected for in evolution. If they have a harmful effect, they'll be selected against in evolution. So in evolution, we're kind of adapting to be better suited to our environment. So mutations are drivers of evolution. So the first Slido poll that we've got today. So if you go on your Slido phone, this should the Slido on your phone. This should flash up. So can you name any diseases that are caused by mutations in our genes? So we've got cancer 1. So these are all your answers that you're flashing in. Cystic fibrosis is another one, very good. So I'll leave this open for about a minute. Has anyone got any other examples? So a lot of people, so the bigger the words are here, the more people are saying them. Down syndrome is not really a mutation. That's caused by having 3 number 21 chromosomes. Sickle cell anemia, yeah, that's caused by a mutation. Lots of people are saying cystic fibrosis as well. A lot of kind of brain diseases have roots in your genes. So for a lot of these diseases, we say that it's kind of the genes interacting with the environment, so they've got kind of a multi-component to their development. So Huntington's is caused by mutations. So Huntington's is a slightly different type of mutation. So at the end of so we've got a Huntington protein, and so a gene is coding for that protein, and at the end of the protein we have a variable number of repeats of a particular triplet of bases. So in Huntington's, the longer the repeat of that triplet of bases, so the more triplets of bases we have there, the higher the chance of you developing Huntington's is. Color blindness is found in our genes as well. So this one here is a good one. Familial adenomatous polyposis coli. So that's a genetic disease which kind of increases your risk of developing colorectal cancer. So you get lots of polyps right from a young age in your intestines. So a lot of people are putting things like allergies, asthma, or eczema. So a lot of diseases have genetic origins or have a genetic component to them. So you might have a genetic predisposition, and then you might have an environmental factor, which then kind of brings on the disease. So well done. That's a lot of answers there. So well done, everyone. So obviously our big answers here. So a lot of people said cancer, a lot of people said cystic fibrosis, and a lot of people said sickle cell anemia. So these are the big ones, which I'd have expected a lot of people to answer with. So it looked like Slido worked well there, so that's good. So moving on. So our first type of mutation, so probably the simplest type of mutation that we can have, is base substitution. We've got a sequence of bases here, so we're calling this here for this example our original sequence of bases. We've got a cytosine, guanine, thymine, so on. And then a base substitution will get a substitution of one of these bases. So here are the thymine on the original sequence has been substituted for an adenine. So a really simple form of mutation. But obviously, if we have a switch of one base in a triplet, we're changing that codon. So a codon is a triplet of bases. So that's the key word that you should remember. And if we're changing our codons, we're changing the triplet code. So we're going to change the amino acid that's coded for by this triplet even when we're only changing one base. But some of these mutations are kind of seen as less harmful because although they are changing one amino acid, it is only one. So sometimes, we're going to look at some other examples of mutations where we'll get a whole host of amino acids changed. So as I mentioned before, this could have a harmful effect. So it could cause a completely different amino acid to be produced, which is going to alter the tertiary structure, the protein produced by that gene. Or because of the degenerate code, which is where we can have multiple different triplets of bases coding for one amino acid. So that's the degenerate code. So that's a key word. So degenerate, that's where multiple triplets code for the same amino acid. So for example, so the CGT and the CGA might code for the same amino acid. So although there's been a change in the codons, both codons might code for the same amino acid. So that would be an example of a silent mutation. So that mutation isn't going to have any effect on the protein produced. So an example of a base substitution that causes a disease is the mutation in sickle cell anemia that causes those faulty red blood cells. So that's a base substitution. So we're switching one base in our original sequence, and then that's producing a mutated sequence. It's got one base different, so we've only got one base different there. So next we're going to look at base insertion or deletion, so we can have both. So this is where we're kind of either inserting an extra base into the sequence, or we're kind of removing a base from the sequence. So we've got the original sequence here, and then this would be an example of an insertion sequence. So here what we've done is we've inserted a thiamine in between the C and the G, the cytosine and the guanine of the original sequence. And in a deletion, we can see here that we've deleted the guanine of the original sequence. So what happens with insertions or deletions, because we're removing or we're adding a base, that's going to cause a frame shift. So we always read DNA from the start to the end. So if we insert a base in, we're causing a frame shift. So we can see that all of the codons after the DNA, after the base insertion, are changed. So that's a frame shift there. So the DNA is read in a completely different way, all caused by the insertion or the deletion of one base. So it's changing every amino acid coded for after the sequence. So if we look at the insertion here, we've changed this codon here, so we've got TAA instead of AAT, so those might code for a completely different amino acid, and then we've also changed this one down here. So we're coding for different amino acids because we're changing all of the triplets after the insertion or deletion. So an example of a mutation that involves insertion or deletion, which causes a disease, is the cystic fibrosis mutation. So the cystic fibrosis gene codes for a protein which is involved in kind of calcium influx and efflux out of a cell. So when we change this, we kind of alter the production of mucus, so we get really thick, sticky mucus, which clogs up the lungs and things like our pancreas and other things. So cystic fibrosis is quite a nasty condition to have to live with. So base substitution and base insertion or deletion, so they're quite simple, so that we can look at them on kind of a base sequence level. So now we're going to look at a couple that are a bit more complicated. So our first example is inversion. So this is where a section of the DNA becomes separated. So we can see here, so this section here has become separated from the original, from the chromosome. It then flips around and rejoins in the same place but in the reverse order. So if we look here, so we've taken this bit from the chromosome, we've removed it, and then we've got this bit that's flipped around and it's reinserted in the opposite in the opposite orientation on the same chromosome. So that's having quite a significant effect over a kind of a multitude of genes there. So if we pretend that each of these different colors is a different gene, this is changing the gene code, the code for the gene code for a variety of different genes. So it's not just so where we looked at base substitution, that's probably only acting on one gene, and same with insertion or deletion, whereas this is affecting a variety of genes. So they can range in size, so we can have only a single codon that kind of is extracted and flips around, or you can have a whole section of a chromosome. So this would be an example of a whole section of a chromosome that's being removed from the strand, it's flipping around, and then it's rejoining the strand, but in the opposite direction. So obviously, the bigger the inversion, the more genes that are affected, the more faulty proteins that are produced, and it's going to have a worse effect on the individual. So we have a variety of diseases that are caused by an inversion type mutation. So severe haemophilia A, that's affecting the clotting of our blood. Infertility, then acute leukemia. So we're going to look at a bit about leukemia later on. So some forms of leukemia are caused by this type of mutation. So that's inversion there. So we're separating a section of the chromosome from the original chromosome here. It flips around and then it's rejoining the same chromosome. So our final type of mutation that we're going to look at is translocation. So like in inversion, in translocation, we're getting a section of a DNA which is becoming separated. And then it's going to insert on another part of the chromosome or on a completely different chromosome. So if we can see here, so this blue chromosome would have been all blue before. What's happened is this section here has become separated, and then it's inserted on this other chromosome, and the same's happened for the red chromosome here. So whereas in inversion, the section of DNA was becoming separated and then rejoining that chromosome, in translocation we get a section becoming kind of dislodged and then attaching to a completely different part of our DNA. So either a different chromosome or a completely different part of the same chromosome. So there are a couple of diseases which are caused by these type of mutations. So translocation Down syndrome. So it's a variant of Down syndrome which is caused by translocation. So this is where Down syndrome is caused by 3 number 21 chromosomes. So in translocation Down syndrome, that additional number 21 chromosome then goes on and attaches to a completely different chromosome. And then certain types of bone cancers, if you look at the genetics of them, translocation mutations can be found. So both inversion and translocation can lead to gene recombination. So these two are examples of recombinant chromosomes. So chromosomes that are composed of 2 different chromosomes. So that's clearly illustrated here by the colours. So we had 4 types. So we had our base substitution, which is when we're substituting one base for another. We had our insertion or deletion. So that's where we're either inserting an extra base into the sequence or we're removing a base from the sequence, and those are going to cause a frame shift, which is going to have an effect on all of the triplets downstream from the mutation. We had inversion, which is where a piece of a chromosome or DNA sequence was becoming separated from where it normally is, flips around, then rejoined back onto where it was before, but in the reverse order. And then we had translocation, which is where a section of the DNA is becoming separated and then attaching to another chromosome. So 4 types of mutations there for you to remember. So we've looked at the different types of mutation there, but now let's look at what can cause a mutation. So one of the potential causes of mutation is an error in replication. So approximately 1 in 100,000 nucleotides is added wrong, and considering the amount of nucleotides that makes up our one strand of DNA, they're relatively common. But the reason that we don't have more mutations occurring and we don't have kind of more cancers, more faulty proteins produced, is because our cells have proofreading mechanisms that occur after the DNA replication stage in mitosis, and they kind of so the cells will proofread the new DNA produced, and if they find an error in it, they might repair that error. So they might remove, if there's been an insertion, they might remove the inserted nucleotide. Or if the mutation's bad enough, so say if we've got a really bad translocation or a really bad inversion, those cells are going to go they're going to undergo apoptosis, which is controlled cell death. So if the error in the cell division is bad enough, our cells have mechanisms which can stimulate apoptosis to prevent kind of a faulty cell with faulty DNA being produced. But some of these mutations do kind of slip through the net. So that's where our mistakes can happen. So we're going to look at cancer later on. So in all of our cells, we have genes called tumor suppressor genes. So these are the type of genes that are involved in this proofreading of the DNA. And in cancer, we can get mutations in the proofreading genes. And obviously, if our cells can't proofread properly, then that's going to give rise to more and more mutations. And those mutations are going to be what causes the cancers. So another cause of mutations is chemical mutagens. So this is why smoking causes lung cancer. It's because of the tar in the tobacco. So what can happen here is that we can get parts of the mutagens can attach onto the DNA, or can remove bits from the DNA, so that the changing of the DNA bases can give rise to mutations. So here we've got an example of deamination. So we're removing the amine group from the base and replacing it with an oxygen. So these are producing completely different bases, and that's going to give rise to mutations. So far we've had errors in replications, we've had chemical mutagens that can alter DNA and cause mutations. And then we can also get radiation. So this is why UV radiation, so excessive exposure to UV radiation can cause skin cancer. It's because radiation can penetrate our cells, and if our radiation hits our DNA kind of head on, that will cause breaks in the strands. And obviously if we've got breaks in our DNA strands, that's where things like inversions and translocations can come about. So both of these two types of mutations rely on breaks in the strands, and then the either flipping in the inversion and reattaching to the chromosome, or in translocations just attaching to a completely different chromosome. So radiation striking our DNA is facilitating inversion and translocations. Or there can also be examples where mutations are introduced during the repair for these strand breaks. So we can either get the radiation hitting the DNA and breaking the strands and allowing inversions and translocations to occur, or the mutations can be introduced during the repair of these strand breaks. So we're going to look now, so we've looked at examples of different types of mutations. We've looked at ways in which DNA mutations can be caused. Now we're going to look at effects of mutations. So the first effect that we're going to look at, or possible effect, is neutral effects. So mutations that cause no changes to the functioning of an organism. So another word for these is silent mutations. They're called silent mutations because they're not having any effects. They're kind of quiet. So they're having no observable impact on an organism's phenotype. And this is the majority of mutations. So the majority of mutations are having no effect on an organism's phenotype. So we're going to look at, so there's a few different ways that a mutation might have no effect on an organism's phenotype. So the first is if the mutation happens in a non-coding region of the DNA. So obviously we have coding regions of our DNA, which is where our triplets are found, which are coding for our amino acids. And then we have non-coding regions, which are altering the expression of the coding regions, and things like that. So they're not coding for amino acids, these parts. So if a mutation is happening in one of these parts, it's not going to have a knock-on effect on the amino acids produced. Another example is what we spoke about earlier, to do with the degenerate code. So that's where we can have multiple triplets of bases that are coding for the same amino acids. So we've got 4 triplets of bases, so we've got 64 possible combinations, possible triplets that we can produce, but we've only got about 20 amino acids, slightly more. So we can see that within those 64 possible triplets, we're going to have some overlap in which ones code for different amino acids. So if I give an example, so ATC might code for the same amino acid as ACG. So that's an example of the degenerate code. So here we've got an insertion. If we think about this triplet, we've inserted a thymine after the adenine. So this is an example of a silent mutation there. So if a mutation is producing a new codon that codes for the same amino acid, that's going to have no effect on the organism. There might be mutations that occur in coding regions, but the changes in amino acids don't affect the functionality of the protein. So if they're occurring in a certain part of the protein, which is kind of less pivotal or isn't kind of a binding region, maybe that might not affect the functionality of the protein. Or a mutation might actually change the protein, but that's not having an effect on the organism. So it's not having a negative effect on the organism. It's kind of just, it's a sideways effect. So an example of that is a blue eyes phenotype mutation. So that's having a neutral effect. So we don't have a survival advantage if we've got blue eyes. It's not causing disease, but that phenotype mutation is having a neutral effect. So it is a mutation, but it's having a neutral effect. So 4 ways that we can have kind of silent mutations. If it happens in a non-coding region, if it codes for the same amino acid, if it doesn't affect the functionality of the protein produced, or if it does change the protein, but that new protein doesn't affect the functioning of the organism. So 4 ways that we can have silent mutations. So now we're going to look, so we've got harmful effects. So they're the second most common. So neutral effects, the majority of mutations are going to be silent mutations, and then some rare mutations are going to have harmful effects. So examples of these are genetic genetic conditions like cystic fibrosis, or then some types of cancer, and these mutations are probably going to occur later on in the lifespan. Genetic conditions you're going to be born with. Cancers are often going to develop later on in life, but we can inherit certain conditions which give us a predisposition to cancer. So we can inherit some mutations which are going to increase our chances of developing cancer further down the line. And then finally, some mutations can actually have beneficial effects. So these are the least common mutations. They're very rare. An example of a beneficial mutation is the Hbc mutation, so a mutation of hemoglobin. So the Hbc mutation is affecting the shape of red blood cells, and they reduce the chance of, and this mutation reduces the chance of malaria. So if we've got 1 allele mutated, you've got a 29% reduction in chance of malaria. If you've got 2 alleles mutated, you've got a 97% drop in your chance of developing malaria. And your side effects are very minor anemia, and they're not affecting your an organism's functioning. So this shouldn't be a condition that causes sickle cell anemia, so the Hbs mutation. So although the sickle cell anemia mutation is thought to reduce the chance of malaria, we would class that as a mutation with a negative effect, because sickle cell anemia is a very nasty condition to live with. So you get kind of clogging up. So you get kind of thicker blood, and it clogs up small blood vessels and causes really bad pain. So we're going to do a Slido quiz now. So we've got 5 questions here that are to do with the content that we've covered so far. So with these Slido quizzes, each question is a lot like a hoop. So there'll be a leaderboard at the end. So the more questions that you get right, the higher up the leaderboard you'll be, and you might have the chance of winning. So this is just a bit competitive to make things a little bit more interesting. So if you get the Kahoot up on your phone, and you need to enter your name at this point. So we've got Lucy in already. So please keep your names as your birth name or a nickname. No rude names. So I'll just give you a minute to enter your name. Just another few seconds. So it looks like no one else is joining, so we'll start the quiz now. There's no time limit on these questions. I'll just move forwards once enough people and once the majority of people have answered. So we're going to go with the first question now. So a gene normally reads, like the sequence there, a mutation's occurred. The mutated gene now reads as followed. What type of mutation is this an example of? There's about 50 people in the quiz, so I can see, and I'll move on once the majority of people have answered. If I move on now, so the correct answer there was base substitution. So you can see here, if we look at the two codes, so we've swapped a cytosine for a guanine in the mutated gene. So well done for everyone that got that one right. So the next question is this. So similar type of question. So we're again just looking at what type of mutation has occurred. Another couple seconds. The correct answer there was a base insertion. So if we look at the mutation, so the first one read CGT, AGC, and the mutated gene, we've inserted a thiamine. So we've inserted another thiamine after the last thiamine, and that's caused a frame shift. So this is an example of a frame shift. So all of the triplets after the insertion have been changed here. And that's because we read DNA, we read DNA in triplets. So if we insert or delete a base, that's going to have a knock-on effect on all the bases downstream. That was a base insertion, was the correct answer there. So another same type of question. Give you a chance to read through it, and then and then you can answer it. Last couple of seconds. The correct answer there was an inversion. So if we see, so what would have happened here is this section of DNA has been cut out or has been removed somehow from the original chromosome. It's flipped around and it's reinserted back where it was. So the mutated gene sequence is the opposite. So it reads from back to front on the normal sequence. That's an inversion. So well done for everyone that put inversion. So final question like this. So exactly the same type of question. We're looking for what type of mutation has occurred between these two genes, these two sequences, sorry. No. That mutation there was a base deletion. So if we look at this first gene, so we're looking here, so it read TCG GAC, and then we've deleted this base, this second guanine base here, and that's caused a frame shift. So we've had a shift in all of the triplets after that deletion. And it read ACT GCC. There must have been a C in the next codon after. So here's our leaderboard. So the quicker you answer the questions, the higher up the leaderboard you'll be. So well done to P or Pia. 4 out of 4, and you've answered all the questions in 59 seconds combined, which is quite speedy. So we've got 21 questions in total spanning this whole presentation. So at the end, we'll have an overall winner. So just to summarize our stuff that we've covered so far about mutations, so they're random changes to the DNA sequence. They're obviously happening by chance because they're random. Then not all mutations are having an effect, and that's due to they might occur in the non-coding regions of the DNA, or due to the degenerate nature of DNA, so the fact that we can have multiple different triplets of bases coding for the same amino acid. There are 4 types of mutations, so we can have base substitutions, insertions or deletions, inversion, translocation. There are a few causes of mutations. They might be caused by errors in replication that aren't picked up by the proofreading mechanisms cell have, or they might be caused by mutagens, so different chemicals can kind of tag on to nucleotides, and that can cause mutations. We have a few types of mutagens, so we can have chemical mutagens, radiation. So if radiation strikes DNA, it can cause strand breaks, and that can cause mutations. So we can either have no effect, a negative effect, or a positive effect on an organism. And the majority of our mutations have no effect on an organism. So we've got a couple of exam questions now that are covering DNA mutations. So our first one, so this is 1A, this is question 1 on your question pack. So why is the genetic code described as being universal? So So we've got a few different words that we use often to describe DNA. So we can call it universal, we can call it degenerate, it's a triplet code, and then it's read from front to back as well. So the universal nature of the genetic code is kind of describing how in almost all organisms on the planet, the same triplets of bases code for the same amino acids. So in all organisms, so if a fruit fly or in a human, a CAG base triplet is going to code for the same amino acid. So that's the universal nature of the genetic code. So in all organisms, the same triplets code for the same amino acids. So the genetic code uses 4 different DNA bases. What is the maximum number of different DNA triplets that can be made using these 4 bases? So here, so if we look at this like probability, so we've got 4 times 4 times 4. That's our expression for how, so we got 4 possibilities at each base. So if we multiply the chances together, we get 64. So it's 64 possible triplets from the 4 different bases. So if we had 5 different bases, we do 5 times 5 times 5. So it's just basic probability. The transcription of a gene produces pre-RNA. So when we've looked at, we've got coding regions of DNA and non-coding regions of DNA. So when we first do our transcription of DNA, the pre-mRNA that's produced, we've transcribed both the non-coding regions and the coding regions. But obviously our non-coding regions, they're not important for making our proteins, because they're obviously not the triplets in the non-coding regions aren't corresponding to amino acids. So then we have a process that removes the non-coding regions from the pre-RNA, and that process is called splicing. So splicing is where, so our pre-mRNA that's produced, we've transcribed both the non-coding regions and the coding regions, and then we have a process that occurs called splicing, which removes the transcribed non-coding regions from the pre-mRNA. So our final mRNA product only contains coding regions, whereas our pre-mRNA contains both coding and non-coding. That's splicing, so that might be a word that you haven't heard before. So the figure below shows part of a pre-mRNA molecule. Geneticists identify 2 mutations that can affect this pre-mRNA. So this has both non-coding and coding regions in this pre-mRNA. So we've got mutation 1, which is occurring in the exon. So exons are our coding regions. So exons coding. And then our introns are the non-coding regions. So mutation 1 is a single base deletion occurring in the exon. Mutation 2 is a single base substitution occurring in the intron. Mutation 1 leads to the production of a non-functional protein. Explain why. So we've got a single base deletion occurring in the exons, so the coding region. So those deletions are causing a frame shift, and that's going to change the DNA triplets that are occurring after the mutation. So if we've got different triplets after the mutation, we're going to code for different amino acids. That's going to alter kind of the hydrogen bonds between the amino acids in the protein. So we're going to get a change in the tertiary structure of the protein, and that might produce a non-functioning protein. So here's the mark scheme for this part. So the mutation changes the triplets, so that causes a frame shift. That's going to change the amino acid sequence. So we're going to be coding for different amino acids. That's going to affect the bonding between the amino acids, and overall that's going to change the tertiary structure of the protein. So the tertiary structure is kind of the 3D shape of the protein. So for example, if we look at an enzyme, if we have a change in the tertiary structure of an enzyme, that might change the shape of the active site of the enzyme. So we've got a different active site of the enzyme. The active site of the enzyme is no longer going to be complementary in shape to the substrate molecule. So it's not going to be able to catalyze the reaction that it used to catalyze, and that's going to be non-functional. So now we've got mutation 2. So we can see that mutation 2 is occurring in the intron, so the non-coding region. So this question is asking, what effect might mutation 2 have on the protein produced? And it wants you to explain your answer. So the most obvious option that I would have gone with is saying that mutation 2 is having no effect on the protein produced, because the mutation's occurring in the intron, so that's a non-coding region, so it can have no effect on the amino acids coded for, so it's not going to have an effect on the protein. So that's that. If you'd have said no, that would have been correct, but then you could have also said yes, so the mutation might have prevented splicing from occurring, and that might have caused faulty mRNA, so you're going to get a different amino acid structure. So there were two ways that you could have answered that. The most obvious way of answering that question that I would have gone with would have been to say that it's the mutations occurring in the intron, which is non-coding, and that's not going to change the amino acid sequence of the protein. It's not changing the code of that section of DNA. So two possible answers for that last question. So now question 2 in your information in your question pack. So the Amish are a group of people who live in America. The group was founded by 30 Swiss people who moved to America many years ago. The Amish do not normally marry people from outside their own group. There's a lot of inbreeding going on within the group. So one of the 30 Swiss founders of the Amish people had a genetic disorder called Ellis van Creveld syndrome. People with this disorder have heart defects, are short, and have extra fingers and toes. So this syndrome is caused by a faulty allele, so it's a genetic disorder. In America today, about 1 in 200 Amish people are born with Ellis van Creveld syndrome. This disorder is very rare in people in America who are not Amish. So this is an example of the founder effect here. So a small group of people have repopulated, and we've started off with a smaller gene pool, and kind of got an increased prevalence of this rare genetic disorder. In America today, there are approximately 1,250 Amish people who have the syndrome. Use the information provided to calculate the current Amish population of America. So if we know that it's told us already that 1 in 200 Amish people are born with the syndrome, so 1 in 200. And then it's told us that approximately 1,250 Amish people have the syndrome. So if we use the ratio, so if 1,250 Amish people have the syndrome and 1 in 200 people, so 1 in 200 of the population have it. So if we use ratios, so if we're multiplying both sides here by 1,250, we get that there's currently 250,000 Amish people. And that's all just ratios. So we know that 1 in 200 have it. So if it's told us that 1,250 people have it, if we times that if we times 200 by 1,250, we get the overall population as being 250,000. The faulty allele that causes the syndrome is the result of a mutation of a gene called EVC. This mutation leads to the production of a protein that has 1 amino acid missing. Suggest how a mutation can lead to the production of a protein that has 1 amino acid missing. So we know that 1 amino acid is coded for by 3 bases, so that's the triplet code. So if we've got 1 amino acid missing, we must have deleted 3 bases. So we must have deleted a triplet of bases. So by saying you've got a loss of 3 bases, that gets you 2 marks. Or if you just say you've got a loss of a base, that would get you 1 mark. So 2 marks, so simple 2 marks there. If you say you've got a loss of a triplet, that's 2 marks for you. So suggest how the production of a protein with 1 amino acid missing may lead to a genetic disorder, such as Ellis van Creveld syndrome. So what's going to happen? So the way in which our amino acids kind of interact with one another, if we've got one amino acid missing, that might change the bonding between the amino acids, so like hydrogen bonds and things like that. That might alter the tertiary structure of the protein, and it might lead to a non-functioning protein produced. So change in tertiary structure, that might need to lead to a non-functional protein. So 2 marks there. So that's us wrapped up on our section about mutations. So we've covered the different types of mutations there and how the mutations occur, and then we've looked at how mutations can affect our genes. So next we're going to move on and look at regulation of transcription and translation. So I appreciate that we've gone on for about 55 minutes there without a break. So I make the time 10:55 now. So just a 5-minute break now to go and grab a snack or something to drink, and then we'll restart at 11 o'clock dead on, and we'll be looking at the regulation of transcription and translation. So just a 5-minute break now. So getting it started again now. So this section will look at the regulation of transcription and translation. So transcription is obviously the production of mRNA from the DNA, and then translation is the production of a protein from the mRNA at a ribosome. So a few learning objectives. So we'll look at transcription factors first, we'll look at the role of estrogen, we'll look at epigenetics and how gene expression can be increased, can be altered through the methylation or acetylation of histones. Look at the epigenetic treatments for disease being developed. Then we'll look at the translation, the role of kind of invariant RNA, so something that you might not have covered yet. So again, it's relevant for all exam boards, so AQA, OCR, and Edexcel. So there's parts of the specification looking at all of this stuff for all of the 3 big exam boards. So gene regulation. So we know that all cells in the body have the same DNA. So we've inherited our DNA from our parents. So we've got 23 chromosomes from our mum, 23 chromosomes from our dad. But different cells do different things. So the fact that we can have different cells doing different things, so our skin cells are completely different to the cells that make up our heart, is all to do with the gene regulation. So it's the way in which genes are expressed within a cell. So not all genes are going to be expressed within a cell. So genes are either switched on or switched off. So switched on means the genes are being transcribed and translated into proteins. Switched off means the genes are not being translated and mostly not even transcribed. So in different cells, different genes are switched on and switched off, and that's all to do with the cell's function, which genes are switched on and which genes are switched off. So the key idea here is that even though all cells carry the same DNA, different cells have different functions. So different cells have got different genes that are switched on and different genes that are switched off. So there are a few different mechanisms by which genes are switched on or switched off. So we can have transcription factors, hormones like estrogen can have an effect, epigenetics, which is we're going to look at is kind of methylation and acetylation, and then we can have inference RNA as well, which we'll look at the end of this section. So this is multiple choice question. So this question won't go towards the quiz. But the question is asking, what are the non-coding regions of DNA called? So we've got a few different options. So just a few seconds to have a go at that. Just a few more seconds. So the non-coding regions of the DNA are called the introns. So exons are the coding regions, introns are the non-coding regions. So first we're going to look at transcription factors as a way in which gene expression is altered. So a transcription factor is a protein that controls the rate of transcription of a gene. So they're binding to a specific part of the DNA sequence, and they're either repressors or activators. So repressors are reducing the translation of a gene by preventing the binding of RNA polymerase, and activators are increasing the translation by aiding the binding of RNA polymerase. So we're going to look at an example of transcription factors at play here. So this is a specific example that you need to learn for your A level. So it's the Lac Operon. So an operon is a small group of genes that all have a similar function. So the Lac Operon is a gene in E. coli bacteria, or sorry, the Lac Operon is a group of genes in the E. coli bacteria which control how the E. coli metabolize lactose. So normally E. coli want to metabolize glucose like most animals, but in the absence of glucose, they want to switch and metabolize lactose and get energy from the lactose. So the Lac Operon is controlling that. But obviously, because the E. coli they prefer the glucose. In most scenarios, the translation of these Lac genes, we don't want that to happen, or the E. coli don't want that to happen because they don't want to be metabolizing lactose. They want glucose. But when the glucose is absent and the lactose concentration is high, the E. coli want to translate these genes so they can gain energy from the lactose where there's no glucose. So we have a few different regions, so we have a few different transcription factors. So the Lac repressor is activated in the absence of lactose and binds after the promoter region and prevents transcription. So the promoter region of an operon is where the RNA polymerase binds. So the Lac repressor is binding to the promoter region and it's preventing transcription. So when there's no lactose, there's an absence of lactose, obviously these genes are useless and it's a waste of energy for the E. coli to be producing the proteins coded for by these genes if there isn't any lactose. So in the absence of lactose, the Lac repressor binds to the promoter and prevents transcription by preventing the binding of RNA polymerase. Then we also have catabolite activator protein, or CAP, and in the absence of glucose, it binds to a site called the CAP site just before the promoter and aids the binding of RNA polymerase, which increases the transcription and therefore translation of these genes here. So obviously, RNA polymerase, so when we want to transcribe a gene, the RNA bases will bind with their complementary bases. The RNA polymerase will come through and it will join, will produce a strand of mRNA from the bases, and then that mRNA strand will go to a protein, will go to a ribosome, and the mRNA will be translated and a protein produced. So two different transcription factors. So we're going to look at the transcription of these genes in a few different scenarios now. So just a reminder, so the Lac Operon is a group of genes in E. coli which regulate the metabolism of lactose. So E. coli prefer to metabolize glucose, but in the absence of glucose, and if there is lactose there, they'll metabolize the lactose instead. So obviously we don't want these genes to be translated when there's glucose around, but we do want these genes to be translated when there's no glucose and there is lactose. So our first example here, so this is what happens if the Lac repressor binds. So if the Lac repressor binds, there's going to be no transcription because of the fact that RNA polymerase is blocking the RNA polymerase moving along the operon. So the Lac repressor binds in the absence of lactose. So obviously there's no point in having these proteins that are coded for by these genes if there's no lactose, because they're controlling the respiration and the metabolism of lactose. So that's the Lac repressor. The Lac repressor binds in the absence of lactose. If there's in a scenario where there's an absence of glucose and a high concentration of lactose, so if there is lactose present, the Lac repressor isn't going to bind, and if there's an absence of glucose, the CAP protein is going to bind to the CAP site, and that's going to aid the transcription of these genes because the RNA polymerase is going to be able to move along these genes and transcribe them. So that's if there's an absence of glucose and there is lactose present. So if there's an absence of glucose, the CAP is going to bind to the CAP site, and that's going to aid, that's a transcription factor, that's an activator, and it's aiding the transcription of these genes by aiding the binding of the RNA polymerase to the promoter region of this operon. So if I just go through the Lac Operon one more time, so I appreciate it's quite difficult. I found it really hard to understand when I was doing A level. So an operon is a group of genes which all have a similar function, and the group of genes are going to share a promoter region. So the promoter region is where the RNA polymerase binds. So that means that they're all normally transcribed at the same time. So they're transcribed together. So the Lac Operon in the E. coli is controlling the metabolism of lactose, and the E. coli, they prefer glucose, but when there isn't any glucose present, they'll switch to lactose as their primary respiratory substrate, and they'll metabolize the lactose. So they'll metabolize the lactose to gain energy. So when there's glucose present, so if there's an absence of lactose, so there's no point in these genes being transcribed if there's an absence of lactose, the Lac repressor will bind to the operator region, or between the promoter and the operator region, and that will prevent the RNA polymerase moving along and transcribing these genes. If there is lactose present and there's an absence of glucose, the cap protein is going to bind to the cap site, and that's going to aid the binding of the RNA polymerase to the promoter, and that's going to increase the transcription of these Lac genes. So the E. coli is going to be able to transcribe the Lac genes, produce the Lac proteins, and that's going to give the E. coli the proteins required to metabolize lactose to gain energy. So E. coli, really simple, so that's why we look at the Lac Operon as a good example of transcription factors. So just a quick poll here in the Slido. So do you understand gene regulation of the Lac Operon? So if we get low scores, I'll explain it one more time. If we get higher scores, then I won't go over it again. So if as many people answer this as possible, then I'll get a good idea about how well I explained it. The average score is 3.8, which is better than I expected. I'm going to go over it one more time because there are a few people that don't understand it, and that isn't good enough. So transcription factors are proteins that control the rate of transcription of a gene. So they bind to a specific part of the DNA sequence, and they're going to alter the binding of RNA polymerase and alter the transcription of the genes. So repressors preventing the binding of RNA polymerase, and that's going to prevent the production of proteins from the genes, whereas activators are aiding the binding of RNA polymerase, and they're going to increase the translation and production of proteins from the genes. So we look at the Lac Operon as a good example to understand transcription factors. So the Lac Operon, so an operon is a group of genes that all have a similar function, and they share a promoter region. So a promoter region is an intron, so it's non-coding DNA, and the promoter region is where the RNA polymerase binds. And when we transcribe, so in protein synthesis, we need to transcribe the genes first. So what happens in transcription is the RNA polymerase moves along the genes and synthesizes a single strand of mRNA, and that mRNA then moves to a ribosome, and the proteins produced at the ribosome from that template. So in E. coli, the Lac Operon is a group of genes that code for proteins that control the metabolism of lactose in the E. coli. So the E. coli, they prefer glucose. So they prefer glucose. But when there isn't any glucose present, they still need something to respire with. So if there's no glucose present, they respire lactose instead. So lactose is a disaccharide, which is a combination of glucose and galactose. So we have a few different parts of this operon which are important. So the promoter region is where the RNA polymerase binds to, and then if it wants to transcribe the genes, it'll move along and transcribe the gene and will produce a strand of mRNA. We have an operator region, which is just after the promoter, and then we have the cap site. So our Lac repressor is a transcription factor, it's a repressor, so it's a transcription factor which prevents the binding of RNA polymerase to the promoter, and that's going to prevent the transcription of these genes. So because the E. coli prefer glucose, because these genes are for the metabolism of lactose, if there's no lactose present, then there's no point producing these genes. So in the absence of lactose, the Lac repressor will bind to the promoter region and prevent the transcription of these genes. We have another transcription factor called the catabolite activator proteins, or CAP, and that will bind to the cap site in the absence of glucose, and it's going to aid the binding of RNA polymerase, and it can increase the transcription of these genes. So in a scenario where we have no glucose, so what the E. coli prefer to respire, and we do have lactose, the cap will bind to the cap site here, and that's going to allow the RNA polymerase to move along the genes here and control and transcribe these genes. So obviously, when there is lactose present, the Lac repressor doesn't bind to the promoter region. So when the E. coli switch to respiring lactose, there's going to be an absence of glucose, so the cap binds to the cap site. The Lac repressor doesn't bind because there is lactose present. The Lac genes will be transcribed, and that's gonna cause the transcription of these genes, and the E. coli will be able to respire and metabolize lactose. So we're going to move on now. So hopefully that I've explained that well enough for everyone. So E. coli are prokaryotes. They're simple, small organisms, but in multicellular organisms, regulation is much more complicated. So in the mammalian genome, so in mammals, there are approximately 1,500 different transcription factors which are encoded in the DNA. So much more complicated than just the two that we saw on the Lac Operon. And the binding sites for the gene tend to be further away. So those promoter regions tend to be not right next to the genes. They might be a bit upstream. So activators are called enhancers in more complicated genomes, and repressors are called silencers. So an example is, in mice, so small mammals, so a gene in the mice is called TBX4, and it's turned on in the development for the back legs, the blood vessels, and the lungs and umbilical cord. So there's several tissue-specific enhancer sites for the different transcription factors from different parts of the body. So here we have the TBX4 gene. So that's so depending on where along this stretch of DNA the transcription factors bind, is going to control where this TBX4 gene controls the development. So if the transcription factors bind here at the hind leg enhancer site, this TBX4 gene is going to be turned on, and it's going to trigger the development of the back legs. If the transcription factor binds here, the lung enhancer site, the TBX4 gene will control the development of the lungs. And then further downstream, if a transcription factor binding at the umbilical cord enhancer site, this TBX4 gene will control the development in the umbilical cord, and the same goes for the hind legs. So whereas in the Lac Operon we had one site, we had a simple operon, we had one site, which was two different transcription factors that were kind of complementing each other. Here, because this TBX4 gene can control the development in a variety of different places, it's quite clever. So during the development of the mice, wherever it's time to develop, so say it's time along the gestation period for the lungs to develop, a transcription factor will bind to the lung enhancer site, and this TBX4 gene will control the development of the lungs. So depending on where the transcription factors bind along the stretch of DNA will control where this TBX4 gene causes development. So several tissue-specific enhancer sites for different transcription factors. So I don't think that you need to learn this example for your A level, but the Lac Operon is important to learn. So we've looked there at transcription factors. So now we're going to look at estrogen, which is very much interlinked with transcription factors. So estrogen is a hormone which is often associated with women. It's found in everyone. So men also have estrogen. And what estrogen does is it can activate specific transcription factors and therefore alter transcription rates within a cell. So we're going to look at the process of how estrogen can alter transcription rates within a cell. So because estrogen is a lipid hormone, is a steroid hormone, it's lipid soluble. So what this means, if it's lipid soluble, it can diffuse straight through the phospholipid bilayer, so the cell membrane. So it can diffuse straight through because it's lipid soluble. So whereas kind of more protein, whereas peptide-based hormones will bind to a receptor protein on the outside of the cell, because estrogen is a steroid hormone and it's lipid soluble, it can diffuse straight through the cell membrane. Once it's inside the cell, it binds to the receptor region of a transcription factor within the cytoplasm of the cell. So it has a cytoplasmic receptor. So whereas some things have receptors on the outside of cells, estrogen has an intracellular receptor in the cytoplasm, and it happens to be a transcription factor. So estrogen binds to the receptor region of a transcription factor. So what this does is it activates this transcription factor in the cytoplasm. So it's going to change the shape slightly of this transcription factor. So now we have an activated transcription factor, and we're calling this the estrogen activator complex. The activated transcription factor then moves into the nucleus, and it binds to the DNA, just like those transcription factors that the Lac Operon were doing, and it allows RNA polymerase to attach and produce mRNA. So it's an activator here, the estrogen complex. So estrogen diffuses through the plasma membrane. It binds to a cytoplasmic receptor. That the estrogen receptor complex is an active transcription factor. It binds to a promoter region on the DNA, and that allows RNA polymerase to attach and then transcribe the genes after that promoter region. So estrogen, when it binds to its receptor, that complex is an activator. It's a transcription factor. So moving on now, we're going to look at a bit of epigenetics. So another multiple choice question. This won't go towards the quiz. So what are the proteins called that DNA winds around? So can you name those proteins? Those proteins are called histone proteins. So DNA winds around histone proteins. That's how DNA is found within eukaryotic cells. So what is epigenetics? So epigenetics is the study of heritable changes in gene expression that do not involve changes to the DNA sequence. They're heritable, which means they can be inherited from mum and dad, and they're changes in gene expression, but they aren't changes to the DNA sequence. So the changes are occurring, if we think of them as occurring kind of in the introns, so non-coding regions, and they're not causing changes to the DNA sequence. So the heritable changes in gene expression. So we're going to look at how the gene expression is changed within epigenetic now. So you might have heard the fact that kind of diet, stress, trauma, they can all affect your DNA. So epigenetics is the way in which these factors that you wouldn't think would be able to alter our DNA are able to alter our DNA. It's all to do with kind of the binding of different things to DNA. So what epigenetics is kind of changing is it's the way in which DNA is wound around those histone proteins. So if DNA is tightly wound around histone proteins, it's not going to be expressed, and we call that DNA heterochromatic, and that's all to do with how it looks under a microscope. So heterochromatic DNA is tightly wound around histones and it's not expressed. So the RNA polymerase can't get in there and it can't transcribe the genes, so that DNA so the genes aren't expressed. If we have loosely wound DNA around the histones, it's called euchromatic, and this DNA is expressed. So the more tightly wound around the histone protein's DNA is, the less it's going to be expressed. So we have euchromatic DNA, which is loosely wound and it is expressed, and then we have heterochromatic DNA, which is tightly wound around the histones and it's not expressed. So two key words there, heterochromatic and euchromatic. So epigenetics is controlled through chemical tags, either directly onto the DNA or onto the histone proteins. So the first epigenetic tag that we're going to look at is DNA methylation. So methylation, so the addition of methyl groups, which are CH3, is a mechanism of epigenetic control. So if we have increased methylation, so more methyl groups added to DNA, the DNA becomes tightly wound around the histone proteins, it's heterochromatic and it's not expressed. So if we have more methylation, the DNA is more tightly wound around the DNA. That's what heterochromatic DNA is, and it's not expressed. So DNA methylation is reducing the expression of genes. On the other hand, acetylation, which is the addition of a C2H5 group, is kind of does the opposite. So the acetyl groups bind to histone proteins, and it causes the unraveling of DNA. So if we have the addition of acetyl groups to the histone proteins, the DNA becomes euchromatic. It's loosely wound around the histone proteins and it is expressed. So the more tightly wound around the histone proteins the DNA is, and if it's more tightly wound, the RNA polymerase can't get into the DNA, can't access the DNA to produce mRNA. The DNA isn't transcribed, so the proteins aren't produced from the DNA. So all gene expression means is it's kind of the extent to which proteins are produced from the DNA. So if we have increased acetylation, DNA unravels, so it's more loosely wound around the histones. That's euchromatic DNA, and it's expressed. So the genes are expressed when we get the addition of acetyl groups. So this type of group up here. So we had methylation. So when methyl groups attach to the DNA becomes more tightly wound around the histones, so it's not expressed. If we had the addition of acetyl groups to the histone proteins, the DNA unraveled and it became expressed. So they have kind of opposite effects and they control the epigenetics. So changes to the epigenome. So the epigenome is kind of is the epigenetic makeup of our DNA. Epigenome can cause cancer. We'll look in more detail about this later. So I spoke earlier, so we've got these genes called tumor suppressor genes, and so if we get a mistake in mitosis, the tumor suppressor genes will recognize those mistakes and will repair the mistakes, or they might cause the death of the cell. So if a tumor suppressor gene is methylated, so it becomes less expressed, those tumor suppressor genes aren't going to function, and we're going to get uncontrolled cell division, which is what cancer is. So a final way that we can control the expression of genes is through the regulation of translation by interference RNA. So what RNA interference is, it's the targeting and destroying of mRNA by small interference RNA. So we haven't just got two types of RNA. We haven't just got tRNA and mRNA. We have other types of RNA. So another example of RNA is small interference RNA. So this RNA interference happens in all eukaryotes and some prokaryotes. So we're going to look at the process now of how small interference RNA can regulate translation. So our first step in this process is that double-stranded RNA is going to enter the nucleus. So we've got a long strand of double-stranded RNA entering the nucleus through the nuclear pores, and then the double-stranded RNA is cut to produce small interference RNA. And the enzyme that's cutting, so double-stranded RNA is entering the nucleus through the nuclear pores, and it's cut straight away to form small interference RNA. We then have enzymes within our nucleus called mRNA cutting enzymes, and they're inactive at the moment. But when they join with the small interference RNA, so when the enzyme and the small interference RNA combine and form a complex, that's going to activate this enzyme. So normally inactive, but when the enzyme complexes with the small interference RNA, we get an active enzyme small interference RNA complex. Then then this complex is able to attach to strands of mRNA in the nucleus, where genes have just been transcribed, and it's able to cut up these mRNA. So that's preventing the mRNA getting out of the nucleus, moving to a ribosome and being translated. So the small interference RNA prevents translation by cutting up mRNA. So just to go over once again what happens. So the double-stranded RNA enters the nucleus and it's cut up to form small interference RNA, or siRNA. The small interference RNA forms a complex with mRNA cutting enzyme and activates the enzyme. The active complex can then attach the mRNA strands and cut up these strands, and that's preventing the translation at ribosomes by the mRNA. So we're preventing the mRNA reaching ribosomes and being translated by the ribosomes. So we've got another quiz now on all the stuff on gene expression and regulation that we've covered so far. So if everyone gets the Slido back up on their laptop on phones, we've got 60 people joined, and we're going to get started. So what does the addition of methyl group tags do to a gene? So we've got a timer for this one. So you've only got 30 seconds to answer this question. So 18 seconds. You need to be quick. Actually, that's not going to work. Sorry, let me just go through and turn off these. I just need to turn off the timer because that doesn't work with the live delay. Okay, so that should work now. Sorry for that. So there we go. So have another go at this. So no longer to have a timer, I'll just move on when enough people have answered. So what does the addition of a methyl group tag tend to do to a gene? So another couple of seconds. It's the same Slido code as before. So the addition of a methyl group tag causes the DNA to wind more tightly around the histone proteins, and that's going to reduce the transcription of the gene. So it's going to switch off the gene. What is the function of the genes in the Lac Operon of E. coli? Another few seconds. So the majority of you answered code for proteins that control lactose metabolism, and that's the correct answer. So E. coli prefer to metabolize glucose, but if there's no glucose present and there is lactose, they'll switch what they want to metabolize to lactose, and those genes in the Lac Operon will be transcribed, and the E. coli will have the proteins available to metabolize lactose. So if the glucose concentration is high, how is the transcription of Lac genes prevented? So the glucose concentration is high and the lactose concentration is low. How is transcription of the Lac genes prevented? So the correct answer there was the Lac repressor binds to the promoter region and prevents transcription. So when we have a high concentration of glucose and a low concentration of lactose, that Lac repressor binds to the promoter region and prevents transcription. So when the glucose is high, you also wouldn't get the binding of CAP to the CAP site, but these two answers were wrong because they didn't really talk about that. So where is the receptor that estrogen binds found in the cell? Few more seconds. So the estrogen receptor, because estrogen is lipid soluble, it can diffuse straight through the plasma membrane and bind to its receptor in the cytoplasm of the cells. So what protein does small interference RNA form a complex with to enable the cutting of mRNA? Okay, so the correct answer there was cutting enzymes. So the cutting enzymes are inactive until it forms a complex with the small interference RNA, and then that complex can go and cut up the mRNA that's produced and prevent it from reaching ribosomes. So remember that splicing enzymes were the enzymes that remove the non-coding regions from the mRNA, from the pre-mRNA to produce the mRNA. So it's not splicing enzymes. So far we've got 3 people that have got all of the questions right so far. So T, Oliver and NASA. So well done to all of you. We've still got a lot more questions left. So just to summarize everything that we've covered in the gene transcription and translation section. So different cells have different genes, have different genes switched on and switched off, and that's what gene regulation is. So despite all cells having the same DNA, in different cells, different genes are switched on and switched off, and that's what gene regulation is. Transcription factors are proteins that bind to specific sites on the DNA. And activators, so one type of transcription factor, increase the rate of transcription. So they'll aid the binding of RNA polymerase, whereas repressors prevent the binding of RNA polymerase and decrease the rate of transcription. In eukaryotes, activators bind to enhancer regions and repressors bind to silencer regions. So we looked at that example in the mice of where the different transcription factors binded, altered which regions of the mice were the gene which we were looking at controlled the development of. So estrogen can activate activator proteins by binding to them. So, and that estrogen activator protein that enters the nucleus and acts as a transcription factors. So epigenetics are heritable changes to the structure, and it does not affect the base sequence. So it's heritable, so they can be inherited, and they're kind of heritable changes in gene expression, which don't involve changes to the base sequence. And it's all controlled by chemical tags on the DNA. And DNA can either be heterochromatic, where it's tightly wound around histone proteins, or euchromatic, where it's loosely wound around histone proteins. And when the DNA is loosely wound, it's transcribed more readily. So methylation is the addition of methyl groups to DNA, which causes the DNA to become heterochromatic, and transcription and translation decreases, whereas acetylation is the addition of acetyl group to histone proteins, and that causes the DNA to become euchromatic, and transcription and translation increases. And changes in the epigenetics can cause cancer. So we've looked at different ways in which the structure of protein, the structure of histone proteins and the structure of DNA, and kind of the direct and how transcription can be increased or decreased. We did look at a way that translation can be regulated. So small interference RNA. So small interference RNA forms a complex with cutting enzymes, and it targets mRNA and cuts it up. So that prevents the mRNA from leaving the nucleus and being translated at the ribosomes. So a few questions now. So this is question 3A and 3B on your pack. So define what is meant by epigenetics. So we've looked at the definition a few times. So it's heritable changes in gene function without changes to the base sequence. So it's all to do with those chemical tags on the DNA, on the DNA or the histone proteins. So in eukaryotes, transcription of target genes can be stimulated or inhibited when specific transcription factors move from the cytoplasm into the nucleus. Estrogen, methyl groups and acetyl groups are control factors that can play a role in initiating transcription. Complete the table to show features of these control factors. Put a tick which shows the correct feature. So estrogen binds with a protein in the cytoplasm, and that complex then enters the nucleus and acts as a transcription factor. Methyl groups bind with the DNA and cause it to become more tightly wound around the histone proteins, so it's heterochromatic and it's not transcribed. Acetyl groups bind to the histone proteins and cause the DNA to become more loosely wrapped around or wound around the histone proteins and aid the transcription of the DNA. So that was the correct boxes that you had to tick there. So the key mistake that you can make is saying that acetyl groups bind with the DNA. Acetyl groups bind with the histone proteins, methyl groups bind directly to the DNA. So this is question 6 now. So figure 1 shows part of a gene that is being transcribed. So name enzyme X. So enzyme X looks like an enzyme that's synthesizing RNA. So enzyme X is RNA polymerase. So 6bi. Estrogen is a hormone that affects transcription. It forms a complex with a receptor in the cytoplasm of target cells. Explain how an activated estrogen receptor affects the target cell. So the receptor then moves into the nucleus, and it acts as a transcription factor. So it stimulates the binding of RNA polymerase, and it's going to aid the transcription of a gene. So the receptor binds to the promoter region, which stimulates the binding of the RNA polymerase, and that causes the transcription of a gene. So estrogen only affects target cells. Explain why estrogen does not affect other cells in the body. And that's all to do with some cells, only some cells have the estrogen receptors present within their cytoplasm. So the estrogen receptor isn't found within the cytoplasm of all cells in the body, only certain cells. And obviously that's all going to be controlled by gene expression as well, isn't it? So that's all of our work on gene expression done. So hopefully we've covered all of these. So we've looked at transcription factors, we've looked at the role of estrogen, we've looked at epigenetics, methylation and acetylation. We've looked at some examples of, we've looked at how a small interference RNA can regulate translation as well. So we say that transcription factors, they are altering transcription, whereas that small interference RNA is regulating translation, so the second part of protein synthesis. So I've been speaking for quite a while there, so I make the time about 11:50 now, so we're going to have a small break now, till about 11:55. So for you to go to the toilet or get a drink, and then we're gonna spend another a bit longer than half an hour before lunch looking at stem cells. So we have lunch at 12:30 for an hour, and then we have an hour and a half after lunch from 1:30 till 3, and we'll be looking at some of the new technologies. So just a 5-minute break now. Okay, if we get started again now. So this section we'll first look at stem cells and then we'll finish off looking at cancer genomics, so kind of the genetics behind cancer. So we're going to look at defining the term stem cell. We're going to look at the differences between the different types of stem cells. We're going to look at some new technologies involving stem cells. Then going to look at the use of stem cells in treating disorders. Then when we move on to cancer treatment, cancer genomics, we'll look at benign versus malignant tumors. Then we'll look at some of the specific genes that are involved in cancer. So we'll look at tumor suppressor genes, oncogenes, methylation, so the epigenetic side of cancer, and also look at a bit about estrogen. So again, this is relevant for all of the different exam boards. So you need to both need to have knowledge about the different types of stem cells, some of their uses, and then also bits about cancer. So we'll cover all of those. So stem cells. So hopefully you're familiar with the term stem cells. So stem cells are unspecialized cells, so they're not differentiated. So that means that all genes are turned on. So genes are turned on. So differentiation is the process by which some genes are turned off. Sorry. So we call stem cells undifferentiated. So one of the key features of stem cells is that they are able to self-renew. So they're constantly dividing, and they divide by a process called oligate asymmetric replication, which means that if we start with one cell and nucleus there, so one cell will become differentiated. So that might say become a skin cell. So that's differentiating, but then we're also producing one more stem cell. So we're starting off with one stem cell, and out of the two daughter cells produced, one will become differentiated. So some of the genes will be switched off and will form a specific type of body cell, and then one of the two cells produced from the division will still be a stem cell. So that's called oligate asymmetric replication. So we're forming one daughter cell. Which we're calling this cell up here, the differentiated cell, and one mother cell. So we're still keeping that one undifferentiated stem cell. So what differentiate means is it's the maturation of a cell. So where some genes in a cell are switched off, and that cell becomes kind of a specialized cell. So it could be a skin cell, could be a neuron in the brain. So that's the process of differentiation, where we're switching off genes in a stem cell to produce a specialized cell. And different stem cells have different abilities to differentiate. So some can differentiate to different extents than others. So the key bits from this slide is that stem cells are undifferentiated. They're unspecialized cells which are able to self-renew because they're dividing continuously and they're producing new versions of themselves. And we have different types of stem cells which have different abilities to differentiate. And differentiate is that process by which some genes within a cell are switched off, and it's kind of the process of specialization. So what are the most undifferentiated type of stem cells called? So what's the name of the most unspecialized stem cells? They're multipotent, pluripotent, totipotent, or unipotent. So this is just a multiple choice question. This isn't going towards your quiz score. So the answer there was totipotent. So we're going to look at totipotent stem cells first. So totipotent stem cells can divide and form any type of cell in the body, and they can also divide to form extra embryonic cells. So they divide to form any body cell, but they also can divide and form and kind of self-renew. So they're known as embryonic stem cells, and they're found in the very early stages of embryonic development. So before the 8-cell stage. So when the sperm first fertilizes the egg and the zygote is formed, those totipotent stem cells are only referring to the first few divisions of that zygote, so where all the genes are switched on. So pluripotent stem cells were kind of the second most undifferentiated. So pluripotent stem cells can divide and form any type of body cell, but they're not embryonic stem cells. So they're again found in the very early stages of embryo development, and they're used to repair tissues. So these stem cells are found just after that 8-cell stage. So we have the totipotent stem cells, which can form any body cells. So they have and also additional totipotent stem cells. So they have all their genes switched on. Pluripotent are again very undifferentiated, so they can divide and form any type of body cell, but they're found slightly later on, still in the very early stages of embryo development, but slightly later on than the totipotent cells, and so they're slightly differentiated. So they're going to have a few genes switched off, but they're still very undifferentiated, and they're not found in the adult body. Multipotent stem cells are the ones that we kind of think of in the adult body. So multipotent stem cells can divide and form certain types of cells. So we have kind of classes of multipotent stem cells. So we can have hemopoietic stem cells. So those that are blood stem cells. So they're found in our bone marrow. So those can divide and form any type of blood cell. So they can't form they can't form skin cells. So we have classes of multipotent stem cells. So hemopoietic stem cells are found in our bone marrow and can divide and form any type of blood cell. Mesenchymal stem cells can divide and form bone, cartilage, connective tissue, and adipose tissue. So kind of our structural tissues within our body. But again, mesenchymal stem cells wouldn't be able to divide and form blood cells. So these multipotent stem cells have slightly more genes switched off, so they can't differentiate and form any cell type. They are slightly limited. So, and then our most differentiated stem cells. So our stem cells with the most genes switched off. But they're still classed as stem cells, are unipotent stem cells. So unipotent stem cells can divide and form one type of cell. So examples of unipotent stem cells are germline cells, which can divide to form sperm only, or skin epidermal stem cells, which can divide and form skin cells only. So unipotent stem cells, it's in the name, they can only divide and form one type of cell, but they're still classed as stem cells. So stem cells are essential for our growth and development. So our growth as adults and as kind of children, but also our development in the mother as embryos. So totipotent stem cells, so our most undifferentiated stem cells with all genes switched on, are the first to form, and they're found in the first few divisions after fertilization. Then pluripotent stem cells form in the blastocyst. So that's one of the stages of embryo development. So that's about 5 to 9 days after fertilization. And pluripotent stem cells can go on to form any of the 200+ cell types in the body when they're kind of stimulated with the right thing. So the right transcription factors switch certain genes off. So pluripotent stem cells are our most important ones because they're found for a while in the embryo and they can divide and form any of the different cell types, whereas totipotent stem cells are only really found before the 8-cell stage of embryo development, so in those first few divisions of the zygote. So if we've just got a table here that's comparing the different types of stem cell, so they obviously range from very potent, totipotent stem cells. They can form every type of stem cell, every type of cell, including themselves, so they can go on to form embryonic cells themselves. Whereas pluripotent stem cells are slightly less potent, multipotent stem cells are lower potency, and then finally unipotent stem cells, because they can only form one cell type, have a very low potency. So the difference between totipotent stem cells and pluripotent stem cells is that pluripotent stem cells can't go on to form extra embryonic stem cells. And then the name kind of gives away the potency to an extent. So totipotent stem cells are found in the early cells straight after fertilization. Pluripotent stem cells are found in the inner mass of the blastocyst and in the umbilical cord. Multipotent stem cells, typical example would be in the bone marrow, so those hemopoietic stem cells, also found in the hippocampus of the brain, spinal cord, and in the amniotic fluid in the placenta. Unipotent stem cells examples include the germline stem cells in the testes that produce sperm, and then the epidermal stem cells that produce skin cells. So zygotes and early stage embryos are examples of totipotent stem cells. Pluripotent stem cells are kind of our embryonic stem cells, and we can also produce induced pluripotent stem cells artificially in a lab. So that's where we switch back on genes from normal body cells, and that will produce pluripotent stem cells. Examples of multipotent stem cells: hemopoietic stem cells in the bone marrow, neural stem cells in the spinal cord, and mesenchymal stem cells, which can go on to produce kind of bone, cartilage, all those tissues. And then I gave some examples of unipotent ones earlier. So stem cells also have extensive medical uses. So our totipotent stem cells are used to replace pluripotent stem cells in diseases like autoimmune diseases or diabetes, or they can also be used to make pluripotent stem cells. We'll look a lot at the uses of pluripotent stem cells, so those induced pluripotent stem cells in the next slide. Multipotent stem cells can be used in treating kind of spinal cord injuries to try and get some regeneration of some of the cells there, and also in the some autoimmune diseases they've been trialed, and unipotent stem cells don't really have any medical uses. So totipotent stem cells are easy to isolate and grow, and they've got unlimited self-renewal because they can divide and reform themselves. But the issue with totipotent stem cells is there's a risk of tissue rejection in the person being treated, and there's also some ethical issues because totipotent stem cells can only be produced in artificial fertilization. So some people would see the use of a sperm and an egg as kind of a waste in the production of totipotent stem cells. Pluripotent stem cells are easy to isolate and grow again, and they also have unlimited self-renewal, but there is a risk of tissue rejection, and if we're just isolating pluripotent stem cells from an embryo, there's also ethical issues. Multipotent stem cells, some of the pros is that there's less tissue rejection because say someone's getting a bone marrow transplant, if they've got certain types of blood cancer, you can match the DNA of those individuals to limit the chance of tissue rejection. And there's also less ethical concerns around multipotent stem cells because we harvest multipotent stem cells from adult volunteers. Some of the cons of multipotent stem cells: they're hard to isolate and they're limited to differentiation. So I've spoken a little bit about induced pluripotent stem cells. So what are they? So induced pluripotent stem cells are made from somatic cells. So somatic cells are normal body cells. So what we do is we can get some somatic cells from a volunteer, and we can switch back on the genes that have been switched off in the differentiation of those somatic cells to reform these pluripotent stem cells. And the positive of these induced pluripotent stem cells is they act in exactly the same way as pluripotent stem cells gained from embryonic stem cells, but we don't have the same ethical issues because we're not having to kind of use embryos in their production. We can also kind of match the induced pluripotent stem cells to the patient by using their own tissue. So say we wanted to make pluripotent stem cells for a particular person, you could use one of their own skin cells and switch certain genes back on that have been switched off in the differentiation of that skin cell, and the pluripotent stem cell produced will be matched to the patient. They'll have exactly the same DNA. And the person that invented the techniques for creating induced pluripotent stem cells won the Nobel Prize. So it's a really significant scientific discovery. So what happens is mitotic stem cells are artificially induced to revert to a pluripotent stem cell state. So they're treated with certain transcription factors which will switch on certain genes that have been switched off in the differentiation of the cell, and that will produce a pluripotent stem cell. So here are some of the examples of transcription factors used. You don't need to learn these, but just know that there are specific transcription factors which are used to switch back on certain genes that have been switched off to reform a pluripotent stem cell. So it changes the DNA packing. So it's gonna change. So they might change how wound around a histone protein a gene is, and that's changing the DNA packaging. So it's gonna switch back on genes that have been switched off. So the medical uses are kind of emerging, and none of the uses of induced pluripotent stem cells are widely used as of yet, but there's quite a lot of promise around them. So at the moment, they have only been kind of tested to treat tissue rejection and skin grafts. So if someone has a severe burn, you can use some of their own skin cells. You can form induced pluripotent stem cells, and then harvest a skin graft in the lab of their own cells, and then use that to attach it back onto their skin and replace the damaged skin. And also in some blood disease treatments, so in leukemia, that they've also been trialed. So let's just look a bit now about what is leukemia. So you probably won't need to know this specifically for your exam, but hopefully it's quite interesting. So leukemia is a type of blood cancer, and it happens where hemopoietic stem cells, so an example of a multipotent stem cell, overproduce abnormal white blood cells, and these abnormal white blood cells then crowd the bone marrow and prevent the stem cells from producing healthy blood cells because they kind of get in the way and they take up too much of the space. Some of the key symptoms include severe fatigue, frequent infections, and that's because we're not producing the right kind of blood cells to fight infections, and also classic cancer signs such as rapid weight loss or swollen lymph nodes are also signs of leukemia. So the diagnosis is often based on blood tests which will indicate leukemia, and then bone marrow tests. So you would insert a needle into the biopsy and into the bone marrow and take a biopsy. So take a small amount of that bone marrow and look at it under a microscope to see if there's leukemia present, and then that can also be used to kind of class what type of leukemia a patient has. X-rays can find swollen lymph nodes, so it can give an idea of where the cancer is spread to, and then tests of other organisms can check for metastasis. So metastasis is where a cancer cells spread from the original site to other organs. So say a breast cancer metastasizing to the liver. So some treatments of leukemia involve chemotherapy, radiation treatment, targeted drug therapy. Then we also have some emerging biological therapies. So these kind of aid the immune system in destroying the cancer cells, and generally what's used is monoclonal antibodies. So these are antibodies produced in a lab which can attach to certain cells and either induce them or destroy them. And if all other treatments fail, then we can have stem cell transplant. So that's where the faulty stem cells in the bone marrow is replaced, and hopefully the patient will be cured of that leukemia. So some other potential uses of stem cells in medicine. So these aren't in treating patients, but they're more in kind of in the discovery of drugs and in the investigation of certain diseases. So stem cells can be used in disease modeling and drug development. So this is based so in early stages of drug development, you test out treatments on single cells or cultures of cells. So induced pluripotent stem cells kind of provide an unlimited supply of patient cells, and we can use those to test out certain drug treatments. In organ synthesis, livers have been synthesized from induced pluripotent stem cells in mice. So we've managed to synthesize a whole organ from induced pluripotent stem cells. Tissue repair. So again, in mice, induced pluripotent stem cells have been used to repair damaged retina, so parts of your eyes. They're also useful in producing cardiomyocytes, so contracting heart tissue. So that's also useful for testing out certain drug treatments on contracting heart tissue. And some scientists have also managed to produce blood type O from induced pluripotent stem cells, and hopefully in the future we'll be able to produce blood from induced pluripotent stem cells, and that will lessen the need for kind of human blood donors, and it will mean that we don't ever have shortages of certain blood types for blood transfusions. So we do have some ethical issues, not with induced pluripotent stem cells, because those we're using adult human cells, but in the use of embryonic stem cells, obviously they're obtained from early stage embryos. So you need sperm and an egg cells, and you fertilize them and you'll produce early stage embryos. And there's some ethical issues around the use of early stage embryos, because some religions or people believe that life starts at conception. So by using and kind of wasting early stage embryos on drug treatments, that's kind of getting in the way of what God would want for some people or religions. So the National Institute of Health in America have created certain guidelines for research involving embryonic stem cells. So the embryo must be created by in vitro fertilization, so not extracted from a mother, must be made in a lab. The embryo may only be used for stem cell extraction when it is no longer needed. So, for example, if a couple is undergoing IVF treatment, if there are some embryos left over at the end of the IVF treatment, that is the only time that embryos are allowed to be used in America. And the donors, so the donor of the sperm and the donor of the egg cells must give consent, because obviously certain people might not want their reproductive cells being used for that type of thing. So before we move on now to look at a little bit about cancer before lunch, I've got a word cloud. So just to get your brains in gear, so where are some of the most common places for cancers to develop? So just as an introduction to our cancer section. Skin cancers, yeah, caused by UV radiation. Breast cancers, lung cancers. So lots of answers coming in now. So you've managed to get a fair amount of the most common places that cancers can develop there. So well done. So cancers can almost develop in any cell type, because what a cancer is an uncontrolled cell division. So it's So the cancer cells aren't responding to the normal cell signaling, and they're dividing uncontrollably. So there are two types of tumours. So we have benign tumours, which we wouldn't call cancerous, and we have malignant tumours, which we call cancerous tumours. And tumours arise due to mutations. So mutations in the tumour cells are going to cause increased cell division, and it's going to cause this tumour growth. So benign tumours, so our non-cancerous tumours, don't spread. So they're encased in the basement membrane. So if we look at this example here, so this is a tissue. So tissues are surrounded by a thing called a basement membrane, which is part of the tissue. And it's kind of forms a capsule around the tissue and makes sure that none of these cells can escape the tissue. So benign tumours don't spread beyond the basement membrane. They have very slow growth. They're not normally life-threatening. And once they're treated, recurrence is rare. But just because they grow slowly and they don't spread doesn't mean that they don't have severe effects. So a lot of brain tumours would be classed as benign or non-cancerous. But because they grow and they push on other brain structures and they get in the way of brain function, they can have very severe effects. They can cause things like epilepsy. You can have increased hormone secretions and things like that. You can have problems with your cognitive abilities. So benign tumours aren't harmless. They grow slowly and they don't spread beyond the basement membrane. So they don't spread to other tissues. Malignant tumours, on the other hand, are cancerous. They are nasty tumours. So they have the ability to invade other tissues and blood vessels, and they grow fast. They release signals which kind of divert blood supplies, and they create their own blood supply. So that's called angiogenesis, where a tumour gains its own blood supply. They spread beyond the basement membrane, so they're not trapped within that capsule. They can spread via the blood or via the lymphatic system. They can be life-threatening because of that spread, and recurrence is common because even if we treat the cancer, there's likely to still be some cancer cells hiding in our body, and at any stage, these malignant cancer cells could divide again and reform a tumour. So the key bits about malignant tumours is that they can spread, they can invade other tissues and blood vessels, they grow fast, and they can spread in the blood to other systems. So metastasis is where cancer cells move from the primary cancer site. So say a breast cancer, they can spread from the breast cancer site into other sites, so maybe the brain, the liver, things like that. So we've looked quite a lot about, we've looked a little bit about cancers kind of invading out of the basement membrane. So malignant tumours invade out of the tissue they're located in. So when cancer surgery is elected for, we need to remove a margin of healthy tissue around the cancer, and that's called the margin. So that ensures that all cancer cells are removed. So by removing some healthy tissues around the cancer cells, so say we've got this tumour here, if we remove the cells in the outline of my pointer there, that's going to ensure that we get rid of all the cancer cells. So that's the margin of healthy tissue. So with the healthy tissues that we remove around the tumour, that's called the margin. So if we have clear margins, so if after we after we look at the tumour that's been removed under the microscope, if there are no cancer cells present within the margin that we removed, the recurrence is less likely of that tumour. But if we do find cancer cells within that margin, the recurrence is more likely. But often, so say in the brain, we can't remove a very large margin around the tissue, because obviously that would cause too much damage to the patient. So if we removed a lot of healthy brain cells, that might affect with the patient's day-to-day living. So it's important for surgeons to be able to know if they've removed all of a tumour, and not at the moment they kind of do that by eye. So they're very skilled and they can identify which cells are tumorous and which cells aren't. But there's certain techniques that are being developed to kind of identify specific cancer cells, and one of these methods is called fluorescent guided surgery. So I'm just going to introduce cancer genetics now, and we'll look at it in a little bit more detail after lunch. So we've got another about 5 minutes before lunch now. So cancer can be caused by gene mutations in a tumor suppressor gene or in an oncogene, or it can be caused by increased estrogen levels or by abnormal methylation of certain genes. So we're going to look at tumor suppressor genes and oncogenes now. So what type of mutation would a tumor suppressor gene undergo in cancer cells? So would they gain function? So would they become more functional? Would they lose function, or would they become neutral? So tumor suppressor genes are those genes which regulate the cell cycle, and they prevent abnormal cell growth. So would they lose function or gain function, or would the mutations in them be neutral in the development of cancer? Tumor suppressor genes lose function, so they have a loss of function mutation in the development of cancer. So I've mentioned it before, but tumor suppressor genes are genes that control mitosis. So they code for proteins which slow down the rate of the cell division. They code for proteins that might check DNA, so check that there haven't been any errors in DNA replication during cell division. And they also, some tumor suppressor genes control apoptosis. So if there's been a big error in the DNA replication, these proteins might cause apoptosis, for the cell to die. So mutations can occur which stop the proteins that are coded for by the tumor suppressor genes being made, and that's gonna lead to uncontrolled cell division. Another thing that can occur is a mutation might occur that prevents the detection of other mutations by these tumor suppressor proteins, and that's going to mean that the cell isn't going to undergo apoptosis when it should be. So examples of tumor suppressor genes are BRCA1 and BRCA2 genes. So mutations in these two genes are linked to breast cancer. So tumor suppressor genes are controlling the cell cycle. They're causing apoptosis in cells where DNA synthesis or DNA replication has gone wrong. So if we have mutations in our tumor suppressor genes, we're going to have uncontrolled cell division. So just final slide before we break for lunch. So oncogenes are genes that make proteins that initiate DNA replication and mitosis. So oncogenes, think of them as our cancer genes. So normally an oncogene will be switched off in the cells. So these effects of oncogenes are kind of bad effects. So we don't want initiation of cell replication, of DNA replication and mitosis, because that's going to lead to that uncontrolled cell division that we see in mitosis. So oncogene mutations can result in genes being permanently switched on, and the result of that is that we're going to get a continuous cell division or uncontrolled cell division. So examples of oncogenes which are switched on in certain cancers are the HER2 gene in breast cancer, EGFR gene in head and neck cancer, colorectal, pancreatic, or the BCR-ABL gene in chronic myeloid leukemia, so a type of leukemia. So tumor suppressor genes in healthy cells are normally preventing cancer formation, whereas oncogenes in cancer cells are causing cancer. So it's 12:30 now, so we're going to break now for an hour for lunch, and then we'll restart at 1:30, where we'll finish off this bit. We'll go through some past paper questions on this section, and then we'll finish with some bits on some DNA, on some genetic technologies, so things like the polymerase chain reaction. So 12:30, So it's 12:30 now, so we'll have an hour for lunch. So we'll restart at 1:30. Okay, so we're going to get going in again now. So the plan for this afternoon is genetics. There'll be some past paper questions and some more the quiz questions on the cancer stuff, and then we'll look at some genetic technology, so things like PCR and gene therapy and things like that. So just a refresher on the key bits of the cancer genetics before we get going properly. So we had our tumor suppressor genes, and these are the genes that control mitosis. So if we have mutations in these tumor suppressor genes, which we're going to see kind of a lack of regulation for our cell cycles. So we'll get uncontrolled cell division, and that is what cancer is. And our oncogenes are mutated versions of proto-oncogenes. There are 40 known proto-oncogenes, and the normal functioning proto-oncogenes initiate DNA replication and mitosis. But if we have a mutation in these normal proto-oncogenes, we get oncogenes, and these can result in cells dividing continuously. So uncontrolled cell division, cancer. So let's just look at now at the role of estrogen in the development of some cancer. So we looked at estrogen as something that can alter gene expression. So estrogen is known to help activate genes, and it's linked to breast cancer in women over 50 and to colorectal cancer. So in premenopausal women, estrogen is made in the ovaries, whereas in the menopause, where our ovaries essentially start shutting down and stop producing estrogen, and then in postmenopausal women, the fat cells produce an enzyme called aromatase, which converts androgens, which are released by part of the adrenal glands, and so this aromatase enzyme converts androgens to estrogen. So postmenopausal women have significantly less estrogen in their system than premenopausal women, and the way in which postmenopausal women produce estrogen is by converting androgens to estrogen. And then certain breast cancers, so the cancerous cells express estrogen receptors. So estrogen actually stimulates their growth. So the treatments for some of these breast cancers that express estrogen receptors is hormonal treatments which aim to reduce the estrogen stimulation. So they might inhibit the estrogen receptors in the cancer cells. So we looked at methylation as a way in which methyl groups bind to DNA, and they increase how tightly the DNA is wound around histone proteins, and that's going to mean that the genes are less expressed. So tumor suppressor genes can become hypermethylated, so they'll become heterochromatic, and they'll be switched off. So the genes won't be transcribed or translated. Whereas oncogenes can become hypomethylated, so they become very readily expressed, and that's going to permanently switch the gene on. So this dysregulation of division can lead to cancer. So hypermethylation for tumor suppressor genes, so that's going to reduce their expression. That's bad because those tumor suppressor genes regulate the cell cycle. So if those tumor suppressor genes are kind of downregulated, the cell cycle is going to go out of control. And the oncogenes, which are the genes that essentially cause cancer, if they become hypomethylated, their expression is going to be increased, and then the cancer might develop. So methylation, that's kind of some of the epigenetic links to cancer. So when we're treating cancer, often a patient's cancer cells, the cancer cells within the patient are sequenced. So we'll look at the specific gene mutations that a patient may have. So for example, if we're looking at breast cancer, the cancer cells might be, the cancer DNA might be sequenced to see if it's expressing estrogen receptors, and then we can target whatever specific mutation that cancer cell has. And if we're targeting each individual's specific tumors, like specifically, then we will probably better to treat the cancer, and we also limit side effects. And certain drugs work better on certain mutations. So this is the idea of targeting therapies. So an example is the HER2 mutation in breast cancer. So HER2 positive breast cancers is where the breast cancer cells have too much of the protein HER2, and this is one of the proteins that kind of controls cell maturation and division and things like that. So if we have too much of this protein HER2, that's going to make the tumor grow. So we can target it with a treatment called Herceptin, and that's going to prevent the tumor growing in response to this protein. So that's an example of targeting therapies. So often cancer, we can find in some cases we can find specific causes of cancer, but often cancers are they arise due to an interaction between genetic predispositions and then environmental factors. So some cancers can be familial, so you'll inherit mutations which it may be your tumor suppressor genes or oncogenes, which might increase the chance of cancer. But then these are examples of some environmental factors that can trigger cancer. So if you were to live in a vacuum and you were never going to be exposed to any of these environmental factors, you would never develop cancer, because your cell's baseline rate of mutation isn't great enough to cause cancer. So it's environmental risk factors like radiation exposure, UV light, radon gas, viruses, air pollution, smoking, kind of red meats in the diet, maybe asbestos in the walls of old homes, are all things that can put chemical tags onto cells and increase the rate of mutation within cells, which might lead to cancer. So we've got some more quiz questions now. So these are all on the stuff that we looked at most recently, so since the last quiz. So I'll give you a little bit less time for these things because I'm conscious that we're running out of a bit of time. So the first question is, what stage of embryonic development are totipotent stem cells found? Another couple of seconds. So it's before the 8-cell stage. So totipotent stem cells are our most potent stem cells. They can become any cell type in the body and also reform these embryonic stem cells, and they're found before the 8-cell stage, so in those first few divisions of the zygote. What type of stem cells are those found in bone marrow? A few more seconds. So the hemopoietic stem cells in bone marrow are multipotent stem cells. What are somatic cells treated with to produce induced pluripotent stem cells? Treated with transcription factors. So we have specific transcription factors which are able to convert somatic cells and able to switch back on genes to produce induced pluripotent stem cells. Which of the following is not a feature of malignant tumors? That was what is they're not, they don't stay contained within the basement membrane. So the membrane that's kind of made of collagen and things like that encapsulates tissues. So malignant tumors are able to breach the basement membrane and spread to local tissues and also enter the blood supply and spread to distant organs. Which of the following would be likely to cause uncontrolled cell division? This is a bit of a harder one. The correct answer there was a hypomethylated oncogene. So if something is hypomethylated, it's euchromatic, so it's being expressed readily. If we have oncogenes, so think of them as the genes that cause cancer. So if an oncogene is being expressed, then cancer is likely to develop, so uncontrolled cell division. So no one's got them all right now. So T is in the lead, 13 out of 14. So a lot of you have only got one wrong. So that's really, really good. Just to summarize our bit on stem cells and cancer. So stem cells are undifferentiated cells which are able to self-renew. And there are 4 key types. So totipotent, totipotent, pluripotent, multipotent, and unipotent. Induced pluripotent stem cells can be produced from adult somatic cells when treated with specific transcription factors, and this has removed the ethical issues surrounding the use of embryonic stem cells. There are 2 key types of tumours, so we have benign and malignant tumours. Benign tumours are encapsulated within the basement membrane, they don't spread, they're slow growing, and they're less life-threatening and less likely to occur, whereas malignant tumours are cancerous. They can invade tissues, they spread via the blood, fast growing, they're often life-threatening, and they can come back if they've been treated. Cancer can be caused by mutations and also epigenetic changes. So cancer mutations may affect the oncogenes, which think of as the genes that cause cancer, and then the tumor suppressor genes, and think of those as the genes that are preventing cancer. So if we have a gain of function mutation in an oncogene, that's going to cause cancer, or if we have a loss of function mutation in a tumor suppressor gene, that could also cause cancer. And then we can also see epigenetic changes at these sites causing cancer. So if we have an increase in the methylation of a tumor suppressor gene, that's going to reduce the expression, and that might cause cancer. Or if we have a loss of methylation at an oncogene, that's going to increase expression, and that might also cause cancer. And an increase in estrogen is also associated with some cancers. And we looked at specific examples of how cancer treatments can target therapy depending on the DNA, the genetics of that cancer. So some examples of genes associated with breast cancer specifically are these ones. You don't need to worry too much about learning the names, but just appreciate there are some genes that give people a predisposition to developing cancer. So I think if you've got mutations in these BRCA genes, that can give you kind of like an 80% lifetime risk of developing breast cancer, so a significant risk. So we're going to go over some past paper questions now to do with stem cells, and also some to do with the bits of the cancer stuff. So explain how increased methylation could lead to cancer. So methyl groups bind to the DNA and they increase the winding of the DNA around the histone protein, and the more wound around a histone protein a bit of DNA is, the less expressed it is, so the less transcribed. So if a tumor suppressor gene becomes hypermethylated, that's going to inhibit transcription, and we might see uncontrolled cell division. And then give one way in which a benign tumor might differ from a malignant tumor, and that's benign tumors don't spread or metastasize. So 3 marks for the first one was a 3-mark question. So methyl groups can be added to tumor suppressor gene. That can lead the transcription of a tumor suppressor gene is inhibited, and that might lead to uncontrolled cell division. And then the part D saying that benign tumors can't spread or metastasize. So SCID is a severe inherited disease. People who are affected have no immunity. Doctors carried out a trial using gene therapy to treat children with SCID. The doctors who carried out the trial obtained stem cells from each child's umbilical cord. Give 2 characteristic features of stem cells. So the 2 key characteristic features of stem cells is that they can keep dividing, so they self-renew, and they are undifferentiated. So they'll replace themselves, keep dividing, and they're undifferentiated. The doctors mixed the stem cells with viruses. The viruses had been genetically modified to contain alleles of a gene producing full immunity. The doctor then injected this mixture into the child's bone marrow. The viruses that the doctor used had RNA as their genetic material. When these viruses infect cells, they pass their RNA to 2 viral enzymes inside the host cells. One of the viral enzymes makes a DNA copy from the virus RNA. Name this enzyme. So normally we'd go DNA to mRNA. That process is called transcription. if we go from viral RNA to DNA, that's called reverse transcription. So the enzyme that makes a copy of the virus, a DNA copy from the virus RNA, is called reverse transcriptase. So HIV is called a retrovirus because it works by this mechanism. So it has reverse transcriptase enzymes that converts the HIV RNA genome into a DNA genome, and then that DNA genome is inserted into the host cell. The other viral enzyme is called integrase. Integrase inserts the DNA copy anywhere in the DNA of the host cell. It may even insert the DNA copy into one of the host cell's genes. The insertion of the DNA copy in one of the host cell's genes may cause a cell to make a non-functional protein. Explain how. So if we insert a strand of DNA into a gene, that might alter the base sequence within that gene, and that might mean that different amino acids are coded for, and the tertiary structure of that protein might change, and it might become non-functional. So it's like this is a bit like an insertion, a base insertion mutation. So if we're inserting a DNA copy into the host cell gene, that might cause a frame shift. So a different sequence of amino acids is going to be coded for, and the tertiary structure is going to be different. So some of the children in the trial developed cancer. How might the insertion of the DNA have caused cancer? So for example, we've looked at examples of genes that are involved in cancer genetics. So it might inactivate a tumor suppressor gene, and that might cause uncontrolled cell division. So if it affects a tumor suppressor gene, that might inactivate the gene and might cause uncontrolled cell division. So the rate of cell division will be increased. 5 of the 20 children in the trial developed cancer. Although the cancer was treated successfully, the doctors decided to stop the trial in its early stages. They then reviewed the situation and decided to continue. Do you agree with their decision? Explain your answer. So we either need to say yes, we do agree with the decision to continue the trial, or no, we don't agree with the decision to continue the trial. So we have options on both sides. So I would have gone and said I don't agree with it, and that's the risk is high of developing cancer. And although the cancer was treated successfully in the first stage, if it's cancerous, then it might come back later and it might not be so treatable. So you can either agree with it or disagree. So some of the options, if you agreed with continuing the trial, was that SCID patients are unlikely to survive anyway. So, and if the cancer that's developed is treatable, then actually they might live for longer. Or no, the risk of developing cancer is too high and it may not be successfully treated in the future. So question 5 now. So explain how the methylation of a tumor suppressor gene can lead to cancer. So that's all to do with if we methylate a tumor suppressor gene, it's going to become heterochromatic, it's going to be tightly wound around the histones, and it's not going to be expressed. That's going to lead to uncontrolled cell division. So describe how you would plot a suitable graph of these data. So if we look at the data, so scientists investigated a possible relationship between the percentage of fat in the diet and the death rate from breast cancer in women from 10 countries. Their data is shown below. So percentage of fat in the diet of the population, and then death rate of the women from breast cancer per 100,000. So you can see we've got some discrete data here, so it's not continuous. We don't have kind of decimal, we don't have 0.1, 0.2, so it's not on it can't be plotted on a straight line sort of thing, so it's discrete data here. So if we've got discrete data, we've got 2 variables, we normally say that we plot a scatter graph. And the key thing with when we're plotting graphs is if this is the axis, so this is the x-axis, this is the y-axis, the independent variable always goes on the x-axis and the dependent variable always goes on the y-axis. So here we plot the fat in the diet on the x-axis, the independent variable, and then the death rate, which is the dependent variable, on the y-axis. So what can you conclude from these data? So it looks like as we increase the fat percentage in the diet, as x increase, so does y. So if we increase the fat percentage of the diet, the death rate from breast cancer seems to be increasing. but there are a few outliers, aren't there? So it doesn't go up and up and up. There are a few points like here, which kind of buck the trend. So you can say that it looks like they're directly proportional. So it looks like there's a positive correlation here. So the higher the fat percentage in the diet, the higher the death rate. Although you can say there are a number of anomalies which would kind of make your conclusion less valid. So some breast tumours are stimulated to grow by estrogen. Tamoxifen is used to treat these breast tumours. So that's one of the hormonal targeted treatments that I spoke about earlier. So in the liver, tamoxifen is converted to an active substance called endoxifen. Figure 2 shows how a molecule of estrogen and a molecule of endoxifen. So use figure 2 to suggest how endoxifen reduces the growth rate of these breast tumours. So straight away we can see that both of them have these aromatic rings, so benzene rings here. Oh, sorry, both of them have these carbon rings. So 6 carbon rings, don't they? So we can say that they're kind of similar in shape. So we can suggest that the endoxifen binds to the estrogen receptor. And if the endoxifen is binding to estrogen receptor, that's preventing the estrogen binding from the estrogen to the estrogen receptor, and that's going to reduce the growth of the tumour. So if I flash in the answer, so we can say that they've got similar shape, so we can see they probably have similar binding sites. So the endoxifen binds the receptor, and the receptor won't be activated. So this is preventing the estrogen from stimulating the cancer cell to grow. That's for 2 marks. So that's our stuff on stem cells and cancer genomics wrapped up. So we've looked at stem cells, we looked at the different types, we've looked at induced pluripotent stem cells, looked at their use in treating disorders, looked a bit about the different types of cancer and the mutations that can occur in cancer, and then we've looked a little bit about the treatments of cancer. So to finish with, we're going to spend the last hour looking at genetic technologies. So these are quite difficult to understand. There are a few different technologies that you need to be aware of for your exams. So we're going to start off by looking a bit more at the genome and non-coding regions, things like that. Then we're going to look at we're going to look quite a lot at a genetic technology called recombinant DNA, which is where you insert a gene from one animal into the DNA of another organism, and then you try and use that for beneficial effects. We're going to look a bit about the ethical, financial, and social issues associated with recombinant DNA technology. We'll look at DNA probes and DNA hybridization, and then we'll look at genetic fingerprinting. So genetic fingerprinting is what's used. So if you see on like crime documentaries when they're looking at we're looking at like crime scene genetics, that's what genetic fingerprinting is. So again, this kind of stuff is in all the different exam boards, so you need to know this whatever exam board you're doing in the summer. It's a fairly big topic as well. So just to start with, just to get you warm, what is genetic sequencing? So this is on Slido again. So which of those do you think is the best definition of genetic sequencing? So genetic sequencing is a method used to determine the order of DNA bases in a genome. So well done, all of you got that one right. So genome project. So what is the genome? So the genome is all the genetic material in a cell or in an organism. So genomes can be sequenced and read, and by sequencing we mean ordering the DNA bases in a sequence, or in a section of the DNA, or in the whole genome. So there are many projects that have been undertaken or are currently in the works that record the sequence the genomes of many organisms. And the use of it's important to sequence the genomes, particularly of humans, because if we know what a normal genome looks like, then we can identify mutations and different genes that might predispose to certain conditions. So it's really important to know the normal genetic makeup of human genomes, so we know what to look out for if we're looking at disease genotypes. So the first whole genome to be sequenced was that of the bacteria Haemophilus influenzae. So that's one of the bacteria that can cause influenza, that's one of the bacteria that can cause pneumonia, and that was in 1995. So we've come a very long way. You might have heard of things like the 100,000 genome projects, which is where the genome of 100,000 different individuals was sequenced. So it was only in 1995 that we first sequenced the genome from a really small bacteria, and now we can do hundreds of thousands of people. So the methods that are used to do this genome sequencing change rapidly. So it was only in 1995 that we first were able to do any sort of genetic sequencing of a whole genome. So up until now, it's a very fast evolving field, and modern techniques are much cheaper, automated and faster than they were before. So the first human genome sequencing was in 2003, and that took 13 years and cost $1 billion, whereas now to sequence the whole genome, it takes 1 to 2 days and only costs $3,000. So you can see the costs are going down, and the time taken is going down as our methods improve. So as we saw with the first ever organism that had its genome sequenced, so the pneumonia causing bacteria, so that's a prokaryotic genome. So they're simpler to sequence and understand because they don't contain introns. So prokaryotic genomes are only composed of coding DNA. So the genome equals the proteome. So the proteome is just the sections of the genome that code for proteins. So think of the proteome as just the exons. So in prokaryotes, the genome and the proteome are the same thing. So sequencing prokaryotic, so as an example like viral and bacterial genomes, helps us identify potential antigens to use in creating vaccines. So at the moment, in the pandemic, they're obviously sequencing all the different variants, and they're using these sequences to try and understand whether different variants spread faster, whether they're more deadly, or whether they respond to viruses. And that's the importance of sequencing genomes. But whereas prokaryotic genomes are quite easy to understand and sequence, eukaryotic genomes are harder because we got the presence of introns, so non-coding DNA. We have regulatory genes, which are kind of regulating the transcription of other genes, and then we have structural sequences, which kind of keep our DNA together. So whereas prokaryotic genome, the genome was the proteome, so all of the genome coded for proteins, whereas in eukaryotes, the whole genome doesn't directly translate to the proteome. So we've got some genes in there that don't code for proteins. So here's all the bits that we're going to look at in genetic technology. So first off, we're going to start with recombinant DNA technology. So the example that's used quite often is the way in which insulin is produced in big fermenters. So when we kind of insert insulin, we insert insulin gene into a bacterial or virus, and then into the bacteria in the fermenters, and then as the bacteria, as they respire and as they grow, they're producing the insulin. So we're kind of altering the bacteria and the fermenters, or the fungi and the fermenters DNA, and getting them to produce something that we want. So we're going to start off looking at recombinant DNA. So there are a few different steps. So we need to isolate the fragment of DNA that we want to insert into another genome, and there's a few methods that we can do that. Then we want to amplify that fragment. So if we've only got a little bit of the fragment, we need much more of it to be able to do with it what we want with it. And that we can either do that by PCR. We normally do that by PCR in the lab. And then we need to insert the fragment into our plasmid or our vector, and then we produce recombinant DNA. And then after we've covered recombinant DNA technology, we'll look at gene therapy and genetic fingerprinting. So three main things that we're looking at. So recombinant DNA technology is the joining of the DNA from two different species. So you might have heard of it being called genetic engineering. So it's used in medicinal treatments, in agriculture, in industry, and also in scientific research. So the first step, as I mentioned, is isolating the fragment. So it's isolating the fragment of DNA that's to be inserted into the original DNA. So if we use the example of insulin, which we'll look at the very end, we need to isolate the human insulin gene, and then we insert it into the DNA of a fungi or a bacteria that's growing in a fermenter. And as the fungi grow in the fermenter, they're going to produce our insulin for us. And that's because we've inserted the human insulin gene into the fungi DNA. So fragment isolation is our first step. So we've got three different methods to do that. So we've got reverse transcriptase, which we looked at a little bit in one of the past paper questions, restriction enzymes, and also this thing called a gene machine. So reverse transcriptase. So reverse transcriptase is an enzyme that catalyzes the formation of a double-stranded piece of DNA from mRNA. So it's a naturally occurring enzyme, and I mentioned it's found in several viruses. So viruses that contain reverse transcriptase are called retroviruses, and HIV is an example of a retrovirus. So they contain the reverse transcriptase enzyme, which catalyzes the formation of a double-stranded piece of DNA from a strand of mRNA. So the first step in this process is that the reverse transcriptase makes a single strand of complementary DNA to the mRNA using free nucleotides that are present within the cell that is invaded. And then DNA polymerase will make a second strand of DNA, which produces a double-strand DNA fragment. So that's our product. So reverse transcriptase, we start off with a single strand of mRNA, and we're able to produce a double-stranded piece of DNA from that mRNA. The main advantage of using reverse transcriptase to isolate the fragment that we want to use is that the complementary DNA is intron-free. So because we're starting with mRNA, which doesn't contain any non-coding DNA, it means that our resulting piece of complementary DNA won't contain any introns. And that's really important, because if we're inserting our fragment into a prokaryote, then they can't process introns, so they can't process non-coding DNA, because it doesn't naturally occur within them. So if we're inserting DNA into a prokaryote, which is what we normally do, into a virus or something like that, then we need to remove the introns so that the prokaryotes can kind of process the DNA we're inserting. Our second method, which is probably the most commonly used method to insert a gene into another organism's genome, is by restriction endonucleases, which are enzymes which are able to cut up a strand of DNA. So again, they're naturally occurring. They can be found in bacteria, and we have different restriction endonucleases have different active sites. So different enzymes are able to cut the DNA at different points. So some enzymes can cut DNA straight through the DNA. So it might go like this, straight through the DNA, and that's creating blunt ends. So if we first look at what sticky ends are, so others can cut DNA at palindromic regions, so where the sequence of DNA reads the same forwards as backwards, and what that does is create things called sticky ends. And then when we're looking to insert our section of DNA into another strand of DNA, the sticky ends will join together. So what a sticky end is if we look at this genome, for example, if we treat, so the whole point of what we're trying to do is we're trying to insert an isolated gene into another organism's DNA. So if we use the same restriction endonuclease enzyme on both DNAs, so we'll cut like this, and this region will read the same forward as backwards. So when we come to, so if we see we've got complementary DNA on both of these sticky ends. So what happens if we then go and insert this piece of DNA into a virus's DNA, and we've done the same thing to the virus, then that's going to aid the virus taking up our isolated fragment, because it's going to have the sticky ends on the virus are going to be complementary to the sticky end on whatever DNA fragment we're trying to insert. So that's what a sticky end is. So it's quite a hard concept to understand. So don't worry too much about it. Our third method of isolating a DNA fragment to insert into another DNA genome is the gene machine, which is the most modern method. So it's using a machine. So what we first need to do, so say we're looking at insulin, we need to identify the amino acid sequence of the protein. And then if we work out the corresponding base sequence for the mRNA of that protein and the DNA of that's coding for the protein, we can then input the DNA sequence into a computer, and then the machine will create small sections of overlapping single-stranded DNA called oligonucleotides, and these small sections are then joined together by the gene machine. So what we're doing in the gene machine is we're starting with a protein. We're working out what the mRNA sequence for that protein and the DNA sequence that the coding for that protein is. We then input the sequences that we know for this protein we're trying to produce into the machine, and the machine will then produce small single-stranded bits of DNA called oligonucleotides, and then it will join these smaller sections together to produce a section of DNA which is coding for the protein that we've started with. So that's fragment isolation. So, for example, if we're doing this process for insulin, that's the way that we're isolating the insulin gene from a human genome. Next, we need to undergo a process of fragment amplification, which is making copies of the DNA fragment. So our first process that we're going to look at is really important to understand, and it comes up quite frequently in exams. So what is the most common technique used to amplify a DNA fragment? So again, this is on Slido. Correct answer is the polymerase chain reaction. So the polymerase chain reaction is the laboratory technique that we use to amplify a DNA fragment. So if we start off with a single fragment of DNA, it's the way that we create lots of copies of that single fragment. So it's done inside a thermocycler machine, and here are some of the ingredients that we need. So we need Taq polymerase, which is DNA polymerase from a thermophilic bacteria found in hot springs. So we'll look at a little bit more about what Taq polymerase is. We need primers, which are short sections of DNA, which kind of attach onto a bit of DNA and act as binding regions for a DNA polymerase enzyme to come in. We then need DNA nucleotides, and we need a DNA fragment to be amplified. So Taq DNA polymerase is a form of extremophile that lives in natural hot strings, so the Taq bacteria. So the important thing about using this type of DNA polymerase is that it can withstand really high temperatures, because the Taq bacteria are found in hot springs. So in this process, we need to increase the temperature of the reaction to about 100 degrees. So we need DNA. So if we were to use our body's DNA polymerase, it would denature if we heated it up to that extent. So we need to use Taq polymerase because it withstands high temperatures. So it's going to be able to do what we want in the conditions that we need to do it. So our first step of PCR is denaturing. So we need to increase the temperature to about 95 degrees, and that's going to break the two DNA strands apart by breaking the hydrogen bonds between the DNA strands. So if we start with two strands and we heat it up to 95 degrees, the hydrogen bonds between the bases, between the strands, are going to break, and then we're going to be left with two single strands of DNA. So that's the first stage. So denaturing. So we heat it up to 95 degrees, break apart the two DNA strands as our first stage. Next, we reduce the temperature to about 55 degrees, and a process called annealing occurs. So this is where the primer is attached. So if we look at this little diagram here, a primer is a short section of DNA which is complementary to one of these strands. So it's going to attach to where we want the part of the DNA that we want to amplify, and it kind of acts as a start point for the other DNA nucleotides to come in and attach to this single strand, and also as a binding site for the DNA polymerase to come and catalyze the formation of a new chain. So far we've had denaturing and annealing. So the third step is synthesizing. So the temperature is increased to 72 degrees, and that's because that's the optimum temperature for the Taq polymerase. The Taq polymerase then attaches to the complementary free so what we've had slightly before this stage is that we get complementary free nucleotides that are in the PCR kind of mixture. So they've attached to the complementary bases on the single strand of exposed DNA, and then the Taq polymerase comes through and catalyzes the formation of a new DNA strand from the single strands that we had after denaturing. So Taq polymerase attaches to the complementary free nucleotides and makes a new strand and two new DNA fragments. So this is basically like artificial DNA replication. So very similar to the process of DNA replication that occurs inside our bodies. So when this process is repeated many times, we even though we started off with a small amount of DNA, it's replicated, just like in our cells, and we'll end up with billions of DNA fragments in automated machines. So we've got three stages. The first stage, which is at 95 degrees, is denaturing. So the hydrogen bonds between the DNA chains break, and we're left with single-stranded DNA fragments. Then decrease the temperature to about 55 degrees, and we have annealing, which is when these short sections of DNA called primers attach onto the single strands, and they kind of act as starting points for other DNA nucleotides to come in, bind. They come in complementary base pair to the exposed bases here. Then a synthesizing, which we increase temperature. The temperature is about 55. It's about 72 degrees. We said in the last slide. And the Taq polymerase enzyme comes and synthesizes another strand of DNA from the single strand that we started with, using the DNA nucleotides that are floating about in the mixture. So some of the advantages of PCR is that it's automated, so it's done by a machine these days. It's rapid, and it doesn't require living cells. So you can take a little bit of DNA. You can find a little bit of DNA in the blood, maybe, or in a cell, and then place it into the machine, and the machine will produce many copies from that single copy that we started with. So PCR is used to amplify a DNA fragment, so to produce many more fragments from a single fragment, and it's also used in forensic science for genetic fingerprint analysis, which we'll look at the end of this session. So PCR is a really important process to understand. So I've got another poll in. So on the scale of 1 to 5, how well do you understand PCR now? So if we get a few low answers, then I'll go over it one more time. So a lot of people are saying 4, but we do have a few 2s and 3s there. So I will just go over it one more time. So we've got three key stages of polymerase chain reaction. So think of the polymerase chain reaction as kind of an artificial way to synthesize new DNA fragments from a fragment that we start with. So it's like artificial DNA synthesis. So whereas in the body, everything we have specific enzymes and we have a process. If we're doing it artificially, we need to use different enzymes, and we need different temperatures. So the aim of PCR is to produce many more DNA fragments from a single fragment that we start with. So they're going to be the same DNA fragments. So we start off with this DNA fragment and we need to produce many more copies of it. So our first step of PCR is that we'll increase the temperature to 95 degrees. And that's going to break the hydrogen bonds between the two DNA strands. And it's going to split the double-stranded DNA fragment into single-stranded DNA fragments. Then the temperature is dropped to 55 degrees and primers attached. So primers are short sequences of complementary DNA, which kind of act as a starting point for other nucleotides that are in the dish to attach to bind to the exposed bases, and they also act as binding sites for the polymerase enzyme, which we're about to look at. So the start of synthesizing a new strands is that primers attach. And then we have the synthesizing stage. The temperature is increased to 72 degrees, which is the optimum temperature for Taq polymerase, which is the DNA polymerase that we're using because it can withstand high temperatures. The Taq polymerase then attaches the complementary free nucleotides in the dish. So remember, we start off with the fragment that we want, the Taq polymerase, primers, and DNA nucleotides. So the Taq polymerase attaches the complementary free nucleotides to the exposed single-stranded DNA fragment, and that produces a new strand of DNA and 2 new DNA fragments. So if we started off with 1 DNA fragment here, split it into 2 single-stranded fragments, and then our end product is 2 double-stranded fragments. And this process occurs billions and billions of times, and we end up with millions or billions of the same DNA fragment, even though we only started with 1. And it's an automated rapid process. So hopefully that clears it up a little bit. So now we've amplified the DNA. What we need to do now is we need to insert that DNA insert the DNA fragment that we have into a vector. So we're probably going to use a plasmid from a virus. So before we do cloning, so before we insert our fragment of DNA into another organism's DNA, we need to modify our fragment to ensure that transcription can occur within our kind of host organism. So what we need to do to modify the to modify the DNA fragment is first add a promoter region to the DNA fragment, which is going to allow RNA polymerase to attach. And then we also need to add a terminator region, which stops the transcription at the end of the gene that we're inserting. So before we move on, so does anyone know what enzyme is capable of joining fragments of DNA together? So gyrase, helicase, ligase, or polymerase? The enzyme that can join DNA fragments together. So double-stranded fragments is DNA ligase. So polymerase synthesizes new fragments, ligase joins fragments together. Helicase can break fragments apart. So now we need to insert our modified DNA fragment into a vector, and a vector is something that will carry the DNA fragment into the host cell. So an example of this might be the plasmid of a virus. So the virus can then infect a host cell and insert the DNA we want to insert into the cell into our host cell. So what we need to do to insert the DNA fragment into a vector is use the same restriction endonucleases that we use to cut the fragment to cut open the plasmid, and that means that we're going to have complementary sticky ends. So we're going to be able to easily insert our DNA fragment into the plasmid. And the DNA fragment and the open plasmid line up at their complementary sticky ends, and DNA ligase will come in and anneal the pieces of DNA together. So stick the two pieces of DNA together. So we need to use the same restriction endonuclease, so we have complementary sticky ends. The sticky ends then line up, and DNA ligase will stick the two pieces of DNA together. So we've started off with the DNA. The DNA is removed from the organism with the desired characteristics, and then a plasmid is also removed from a bacterial cell. The gene of the desired, so we've done all these processes. The gene of the desired characteristic is cut from the DNA using restriction enzymes, and at the same time the plasmid is cut open with the restriction enzymes, and then the cut ends or the sticky ends from both the plasmid and the DNA strands attached together using DNA ligase. Now we need to insert our vector into the host cell. So we do this through a process called heat shock. So first we need to add lots of calcium ions to the mixture, and then we'll suddenly increase the temperature. And what that's going to do is it will make the membrane, the plasma membrane, more permeable, and that's going to allow the plasmid to enter the host cell. So we've got our modified plasmid, we've heat shocked it, and the plasmid has entered our host bacterial cell. So once we think we've been successful in inserting our plasmid back into the bacterial cell, we need to actually identify the cells where we've been successful. So one method that we can use to do that is the antibiotic resistance marker. So this is a bit complicated. I don't think it's that widely used. So what would happen here is the DNA fragment would be inserted into a plasmid that contains two antibiotic resistant genes. So if the fragment is inserted into the tetracycline resistant gene, and then the bacteria are grown on agar, and that's going to produce colonies of bacteria. Then a sample from each colony will be placed on a plate with ampicillin, which is type of penicillin, and only the colonies that survived the penicillin, that survived the ampicillin, are grown in the tetracycline. And we know the colonies that survive the antibiotic do not have the gene of interest. So another more common method that we use to identify transformed cells, which is the method that's probably going to come up in your exams, is the use of fluorescent markers. So the plasmid, so we've already inserted a gene into the plasmid called the GFP gene from a jellyfish, which is the green fluorescent protein. So this gene is going to create a protein that lights up under UV light. So the DNA fragment is inserted into the middle of this fluorescent gene. So if the bacteria take up the gene of interest, that's going to affect the functioning of the gene, and the gene and the correctly transformed bacteria won't light up because we're disrupting the gene which normally glows under UV light. That's the use of fluorescent markers to identify our transformed cells. Then finally we can use enzyme markers, which are again not so common. So the plasmid has the gene for the enzyme lactase, and so we'll insert our desired gene into the plasmid lactase gene, and we can test for the lactase gene. So any bacteria that we've inserted the desired gene in the correct place and correctly won't make lactase, because we've inserted the gene into the middle of the lactase gene, and that's going to affect the functioning of that gene. And then finally, once we have our bacteria, which we know for sure have been modified, they're going to be grown in fermenters, and that's going to produce large colonies of bacteria which are going to produce the protein from our modified inserted gene. So I've spoken a little bit about the example of production of insulin. So the bacteria are genetically modified with the recombinant gene for human insulin, and as the bacteria grow in the fermenter, they'll produce our insulin for us. So our final step, bacteria reproduces, producing millions of copies of identical bacteria, and they're all going to have the desired gene that we. So some advantages of DNA recombinant DNA technology include that it may improve food production. It's good for producing vaccines and also other medications. Some disadvantages is that it goes against the laws of nature, so it's a very unnatural process, or some people might have religious beliefs against it, have the potential for DNA data from people to be stolen. Some people are concerned about the effects of genetic modification on people and the environment, and then some people are worried that we'll use similar technology to make designer babies with desirable characteristics. So we've covered all of our bits on recombinant DNA technology. So we're just going to have a 2 or 3 minute break now, so you can go get a drink before we crack on with our final parts on gene therapy and genetic fingerprinting. So just a couple of minutes now. So I make the time 2:33. So we'll start again in a couple of minutes. So about 2:35 or 2:36. Okay, so if we get going with the last kind of 25 minutes of today. So now we're going to look at gene therapy, a bit on DNA probes, and then after we finish that, we'll look at genetic fingerprinting. So gene therapy is the new and experimental treatment that uses recombinant gene technology to treat or prevent disease. So if we're looking at genetic disorders, so the mutated gene that's causing disease can be isolated, and then a non-mutated version of that gene is then introduced into a vector, so we're looking at is what we've just covered. So the recombinant gene technology, the virus that contains the normal copy of the gene is injected into the patient, and then the patient's cells might take up the wild gene, and the disease might be cured or the symptoms reduced. So it sounds very good, but in practice it's very hard to do. So there's been several successes so far in treating disease such as severe combined immune deficiency, haemophilia, some sorts of blindness and leukemia, and there are a lot of clinical trials going on for many different diseases using this gene therapy technology. So DNA probes are short single-stranded pieces of DNA that are labeled radioactively or fluorescently, and they're used to find sequences of DNA. So the probes attach to another single-stranded piece of DNA, and that process is called hybridization. So say we were looking for this gene here, so CAG TAC TTA. So this is the gene we're trying to find. So if we were to make a DNA probe for this gene, we'd make it would be complementary. So we'd use the complementary bases. And then this probe we've got here would have a fluorescent label attached to it. And if the DNA probe attached to the complementary piece of DNA in the genome that we're looking for, then the fluorescent label would light up, and we'd be able to identify our piece of DNA that we're looking for very easily. So the primary use of DNA probes is to locate specific alleles within genes, and that helps us find cases of heritable conditions, genetic health risks, and likely responses to drugs. So if we were trying to find a specific allele that causes cystic fibrosis, we'd make a DNA probe for that allele. We'd inject it into the host cells, and if the individual had the faulty allele for cystic fibrosis, the DNA probe would fluoresce, and we'd know that individual has the faulty allele for cystic fibrosis. That's the way in which genetic diseases are identified. So personalized medicine is a method of treating patients based on which bases kind of the treatments and the decisions around the treatments into the patient's individual predicted response or risk of disease. So we use the patient's DNA and their genes to predict what diseases might arise from their genes or their response to the treatment. So people will have their whole genome sequenced and discover if they have genetic predisposition for certain genes, and if they know that they have a predisposition for certain genes, they might make lifestyle changes, or they might undergo certain treatments which might lessen their chances of developing the diseases that are predisposed for in their genes. And it can also, another important reason to sequence someone's whole genome is to determine what mutation is causing a disease. So often diseases have several mutations that can cause them. So some of the diseases of the brain can have like hundreds of different mutations which are linked to the disease. So if we know the specific mutation in a certain individual which is causing their disease, we can target the treatment towards the mutations an individual is experiencing. So whenever we're trying to identify, or we're using a patient's genes to make decisions or identify certain genetic conditions, this concept of genetic counselling is really important. So genetic counselling is discussing with patients who have a genetic disease about the chance of passing their inherited conditions on, and it allows them to make informed decisions about genetic testing and IVF. So two people that are both carriers of cystic fibrosis might choose to have IVF and pre-implantation genetic diagnosis to make sure they don't pass on cystic fibrosis to their offspring, or the knowledge that they're both carriers might mean that they choose not to have children, for example. So that's genetic counselling. So we've looked at DNA probes, so we're going to finish off and look at genetic fingerprinting, which is used by the police to try and it's used by the police to identify kind of criminals. And it's also used in things like parental testing. So if the father of someone is unsure, you can use genetic fingerprinting to identify the father. So genetic fingerprinting involves first collecting a DNA sample from two individuals, secondly extracting DNA from the sample, and then the DNA fragments that we extract are then amplified by PCR. And then finally, we want to digest the DNA sample, so restricting endonucleases cut the DNA into smaller fragments just below these things called VNTRs start. So VNTRs are variable number tandem repeats. So these are found in the introns, so the non-coding regions of DNA, and they're long sections of DNA with repeating sequences. So the probability of two people having exactly the same variable number tandem repeats is very low. So it makes an excellent way to identify people. So variable number tandem repeats are non-coding regions of DNA where there's lots of repeating sequences of DNA, and they're used to identify people because there's a very low chance that two people would have exactly the same number of tandem repeats, essentially. So they're mainly used in forensic science to determine who was at the scene of crime. So if we've got a bit of DNA from a crime scene, if we collect the DNA from a suspect, we can compare it with the DNA at the crime scene to confirm whether they were there or not. And that's using the number of tandem repeats in both samples of DNA, and you can use it as a paternity test to find out the father of a child. So variable number tandem repeats are kind of are passed on by both the mother and the father. So depending on how similar two people's variable number tandem repeats are, you can use that to work out whether they're related. So after we've done these three steps, after we've collected the sample, extracted the sample, and cut the sample at where the variable number tandem repeats start, we then need to separate the samples. So we separate DNA samples using a method called gel electrophoresis. So if I flash up the diagram here, so what happens is DNA is loaded into these wells at the bottom. And different samples again go in separate wells. We place a gel over a gel that kind of buffers the liquid, and an electric voltage is applied. So at the top here is positive charge, at the bottom here is negatively charged, and we've got a voltage. So we've got a flow of voltage across this electrophoresis plate. So DNA is negatively charged, so the samples are going to move towards the positive electrode at this end of the electrophoresis plate. So the smaller variable number tandem repeats travel faster and further compared to the longer variable number tandem repeats. So the lower the mass of the variable number tandem repeats, the faster it will move and the further it will move. So that creates kind of separation. So that means that the variable number, so the samples then separate depending on the length of the variable number tandem repeats. And then finally, an alkaline solution is is added to separate the double strands of DNA. So we're left with something that looks a little bit like this. So after we've done the process of electrophoresis, so after the samples have been separated depending on the length of their tandem repeats, the sample then undergoes a process called hybridization, which is where DNA probes are added to the variable number tandem repeats to identify where they are. So at the moment we can't see where the different bands are, but after we've added the DNA probes, the bands show up. And that's after a process of development where the hybridized DNA samples are transferred onto a nylon sheet, and that's called southern blotting, and they're exposed to X-rays or UV light, which will produce this image. So the DNA probes attached to the samples of DNA, and when we shine a UV light on them, the DNA, the variable number tandem repeats will show up because of the DNA probes attached. So we get this image with bands, and we can compare the sample from the, so if we're looking at crime scene, we can compare the sample found at the crime scene with the suspects, and we can see here the sample has exactly the same genetic makeup as suspect 3. So from this you can conclude that suspect 3 and the sample share the same DNA, so suspect 3 was probably the person who did this crime. So just a few final quiz questions to wrap up today. So these are all going to be on the genetic technology section. So what do prokaryotic genomes not contain? The correct answer there was prokaryotic genomes don't contain introns, so they only contain coding DNA. What does the reverse transcriptase produce from mRNA? So tRNA, small interference RNA, single-stranded DNA, or double-stranded DNA? The correct answer here is single-stranded DNA. So if we remember, the reverse transcriptase produces a single-stranded DNA fragment from the mRNA, and then DNA polymerase comes through and converts this and then synthesizes another strand. So overall we get double-stranded DNA, but the reverse transcriptase produces single-stranded DNA from the mRNA. What is the purpose of raising the temperature very high in the first stage of PCR? correct answer there is that it breaks the hydrogen bonds between DNA strands. What enzyme is used to cut open plasmids at complementary points to the fragment that is to be inserted? The correct answer there is restriction endonucleases. So they cut the DNA that we're inserting and the plasmid at complementary points called sticky ends, and that allows the DNA fragment to be inserted into the plasmid. Then what is a DNA probe? So a DNA probe is a short single-stranded piece of DNA that's used to find specific sequences of DNA. So for example, it might be used to identify whether an individual has a certain allele that causes a genetic disease. So what is the name of the sections of DNA that are used in genetic fingerprinting? So like paternity tests and crime scene investigations. Correct answer there is variable number tandem repeats. So here's our leaderboard. So well done. 3 of you managed to only get 2 wrong across the whole day. So well done, everyone. Thank you for taking part. So just to summarize what we've done on genetic technologies. So a genome is the whole of the DNA in a cell of an organism. Sequencing a genome is determining the order of bases in that DNA. Many organisms have had their genome tested. It's important because sequencing genomes can be used to find sequences of proteins, and that might be used in vaccine development. And these days, sequencing methods are always being developed, and they're now very automated, so it's cheaper and faster than it's ever been. So recombinant DNA technology involves the transfer of fragments of DNA from one organism to another. And DNA fragments can be isolated using reverse transcriptase, restriction endonucleases, or gene machine. The DNA fragments are then amplified either in vitro or in vivo techniques. So in vivo is inside an organism, in vitro is in a lab. So PCR is the key amplification method that denatures the double-stranded DNA. Then primers anneal, and then replication occurs, and that's using Lac polymerase. And then in vivo cloning is inserting the DNA fragment into the plasmid of a bacterial cell. That's our final step in recombinant DNA technology. So some of the benefits of recombinant DNA technology is that it might improve food production, vaccines, or medications. Some disadvantages include people being concerned of the effects of GM on people and the environment. So gene therapy is a medical technique of replacing mutated DNA in people. DNA probes are short, single-stranded, and are used and are labelled fluorescently and are used to identify specific sequences in DNA. Genetic fingerprinting is a method of identifying someone by their unique variable number tandem repeat pattern. It's in forensic science and also for paternity tests. So we'll just go through one past paper question before it's 3 o'clock, and that's when we end. So first question, so what is a genome? So genome is all of the DNA within an organism. Chromatin immunoprecipitation is one method used to determine where a transcription factor binds to DNA. The principle behind this procedure is shown in the chart. So first step, add chemicals to bind the transcription factor to DNA. Break down DNA into small fragments and add an antibody specific to the transcription factor. Third step is to remove the precipitated DNA and add the chemical to remove the antibody with the attached transcription factor. And the third step is to amplify the DNA, which will correspond to the site occupied by the transcription factor. So explain why the antibody binds to the transcription factor. And that's all to do with the fact that they're complementary in shape. So get a mark for saying that the transcription factor and the antibody have the transcription factor has a specific tertiary structure, and another mark for saying that the two have a complementary shape. So use the chart to explain what precipitated DNA consists of. So far we've got transcription factor, we've got DNA, we've got small fragments of DNA, and we've got an antibody. So the precipitated DNA contains all of those things. So DNA, transcription factor, and antibody. So soybeans are used in a number of processed foods. However, soybeans contain a protein known as P34 that causes an allergic reaction in some people. Scientists have created transgenic soybeans that produce single-stranded C complementary DNA, which prevents transcription of P34 gene. They use recombinant plasmids as vectors to transform soybean cells. After they had screened these cells for production of the P34 protein, they cultured the transformed cells to produce soybean plants. Suggest how single-stranded cDNA could prevent the transcription of the P34 gene. So here, if this complementary DNA binds with the P34 gene, that might block the transcription. So it's going to bind to P34. So that's because it's a suggest question, there are a few answers that could be right. You could say that it binds to the P34 gene, or it binds to a transcription factor, or to a promoter. So suggest questions, they have a few different answers that you could say that are all right. So E, describe the roles of two named enzymes used to insert DNA fragments into plasmids. So here I'd go with restriction endonucleases, which cut the plasmid and cut the DNA fragments at complementary points to create those sticky ends, and also DNA ligase, which joins the DNA to the plasmid so they anneal together. So 1 mark for restriction endonucleases and its role, 1 mark for DNA ligase and its role. So F, the soybean cells were screened for the presence of the P34 protein. This process involved the use of gel electrophoresis to separate proteins extracted from soybean cells. Suggest 2 features of the structure of different proteins that enable them to be separated by gel electrophoresis. So if we remember, gel electrophoresis was where we loaded the things into wells at the bottom. They have a gel which allows them to move. There's an electric voltage across the thing, so we have a positive charge at the top here, positive electrode, negative electrode at the bottom. The things that are loaded into the wells are all negatively charged. They're attracted to the positive charge, and the smaller fragments that are loaded into these wells will travel fastest and travel furthest so they'll travel the furthest up the sheet. So two features of the structure of the different proteins that enable them to be separated by the gel electrophoresis might be the charge or the mass, the different masses. You can say that R groups differ, which is why they have different masses. That was question 7. So that's 3 o'clock now, so I think the time is up now. So I hope everyone enjoys the rest of their day in the sun. Thank you for coming.